gracing us with their presence. I would also like to extend a warm welcome to our chief guest, Dr. Sripat Krishna Devalla, and our judges, Mrs. Sumati Mishra and Mr. Shubham Anand, for being a part of this national level online competition today. I would also like to extend a very warm welcome to Dr. Joshi Manisha S., Head of the Department of Medical Electronics Engineering, Dr. Vijayalakshmi K., Professor of the Department of Medical Electronics Engineering, the faculty members, and all the contestants present here. We are truly elated and honored to have you all here. With expertise spanning in physiology, biology, healthcare, health informatics, mechanics, and engineering, biomedical engineers can combine their diverse skills to create solutions to continuing worldwide health issues, helping to change how patients are treated, lowering the cost of care, and many other amenities. With technology advancing and the demand for cutting-edge medical equipment and devices expanding, biomedical engineering is a rapidly growing field. Owing to this, the Department of Medical Electronics and Engineering, in collaboration with BMSCE, IEEE SP, has come up with this national level online project competition called Biomed Technophilia 1.0. And we are gathered today to witness various teams present their innovative projects that could bring a change to the society. Before we begin the contest, I request all the dignitaries to officially inaugurate Biomed Technophilia 1.0. Thank you, everyone. With this, let us proceed with the inauguration of the competition. I would like to request the head of the Department of Medical Electronics Engineering, Dr. Joshi Manisha ma'am, to formally welcome the gathering.
Dignitaries of the today's event, Principal Dr. Muralidhara S., Dr. Suresh, Vice Principal Admin, Dr. Bhimsha Sir, Vice Principal Academics, Chief Guest of the today's event, Dr. Sripat Devalla, Co-founder and CTO of Origin Health, judges, Mrs. Sumati and Dr. Shubham Anand, deans, HODs of various departments, IEEE student branch faculty coordinator and students, my colleagues in the department and participating teams and their faculty members. Good morning and hearty welcome to all of you for the inauguration function of national level project competition, Biomed Technophilia. This is first edition of this event. To motivate the students in order to take various project works in this area in the healthcare engineering and to create indigenous products is the main objective of this workshop. I wish all the best to participants and good luck for this event. Thank you.
Thank you, ma'am, for addressing us with your kind words. I now request Dr. Vijayalakshmi K, ma'am, to brief everyone about the competition and the concept behind it.
Hi, good morning, everyone. Once again, I welcome all the dignitaries of the event as well as student participants and faculties. I, Dr. Vijayalakshmi K, a faculty coordinator, would like to just introduce about the event participant and registration. We have an overwhelming response from various states of Karnataka. We have around 25 teams and 85 participants. We have 3 software projects and 22 hardware projects. And we have participants from Kerala, Gujarat, Tamil Nadu, Andhra Pradesh, and Karnataka. This is just about the introduction of the registration of the event participants. Thank you one and all. Once again, all the best to all the teams for the presentation. Thank you.
Thank you, ma'am, for sharing the details with all of us and walking us through the event. I now request our Vice Principal Admin, Dr. Suresh Ramaswamy Reddy, to share a few words.
Hello, good morning. Am I audible? Hello.
Yes.
Good morning.
Yes,
Am I
sir.
audible?
Yes, sir. Yes, sir.
Respected chief guest for today's function, respected principal, my esteemed colleagues and participants from different states. Good morning. This is the first time I'm seeing medical electronics is coming with a novel idea of this technophilia. So it's heartening to note that there are about 80 participants from different parts of the country. This is the first of its kind. I'm very curious to see what kind of projects this kids presenting for the next maybe about 12 hours. So I wish each and every participant a success. And one point, success and failure are the stepping stones. Don't worry about failure. So presenting a good project that is what everyone looks for. Thank you very much. So God bless.
Thank you for your constant guidance and support, sir. I now request our Vice Principal Academic, Dr. Bhimsha, sir, to address the gathering. Due to unforeseen situations, the Vice Principal couldn't join us today, but he sends across his best wishes. I now would take the opportunity to introduce you all to our chief guest for the day, Dr. Sripat Krishna Devalla. Dr. Sripat is the co-founder and chief technology officer at Origin Health Incorporated and an entrepreneurial scientist with more than 8 years of experience in artificial intelligence and computer vision. Not only this, he is also developing artificial intelligence technologies to assist in interpreting pregnancy ultrasound examinations. He has also worked with leading clinicians and health systems in Singapore, NHS in the UK, and India to translate deep learning algorithms to simplify clinical diagnosis and management of ophthalmic diseases. I would like to request Sir to share a few words with the gathering present here today.
Thank you for the warm introduction. Good morning to one and all present here. It's an honor to be here today amidst this esteemed gathering, principal, vice principals, head of the department, faculty, department staff, and the judges. I'm also delighted to be a part of this event amidst some of the brightest minds who would grow to be the future entrepreneurs, biomedical engineers, and scientists. I'm an electrical and biomedical engineer by training as well. And so I can relate to each and every one of you present here. I've gone through a similar journey as well. Biomedical engineers are always at the forefront of the greatest scientific discoveries that have been the basis for survival and progression of our civilization in modern times. Be developing life-saving medical devices such as pacemakers, implants, vaccines, disease management, hospital technologies, medical software, and the list goes on. These innovations have changed the way how we live, improved the quality of our lives, and significantly extended our lifespans, reduced mortality and suffering for mankind. Until recently, we've all heard these motivational lines while joining our undergraduate or postgraduate programs, or through marketing materials and brochures we typically see from universities. And all of us have been fallen for these motivational lines and have been deeply motivated. But this lasts probably only until we start to experiment, we start to experience our experiments failing, or we don't land our dream internship or jobs in core biomedical engineering companies. And what happens after that is, people decide to give up on this field and go do something else. No disrespect to people working in pure software or any other field, but my point is simply on biomedical engineers who have chosen this path, right? But COVID has changed things. The world has witnessed firsthand what's truly possible by this group of individuals when they set out to solve some of the hardest problems affecting humanity. This group has engineered vaccines, developed diagnostic kits, robots, medical devices, and several medical technologies in record times. Right now, as a result of this, solutions that typically used to take years for conceptualizing, development, and validation have been ready in span of just a few months. So, how are they able to do this? Right. So they were able to do this because of their interdisciplinary approach to problem solving that involves their interest and passion in medicine, science, engineering, and technology. And I'm sure a lot of you participating or attending today's biomedical technophilia event are here because of a passion for biomedical engineering, medical electronics, and biotechnology, and will go to achieve the same as well, like all the other fellow biomedical engineers. That doesn't mean that we need another COVID scenario for you to prove yourselves. You can do it right now also, without expecting another pandemic. But what's more important for you in this is do not let your scientific curiosity, the spirit of innovation and problem solving, and the inquisitiveness that you have for doing these projects and coming for contests like these die with your final year projects or with your theses or with your graduation. My wish is that they should continue for every one of you, the passion that you have for this field. Irrespective of the results today, I urge each of you to continue your interest in the space by publishing your works in top journals, presenting at top conferences, or even patenting your works if you intend to commercialize something later. But while you're doing this, I want to give you some caveats that your manuscripts that you submit might not be accepted for the first time and get rejected 1 to 10 times. You might not get talk in conference, you might not win contest prize, but it doesn't matter. Focus on learning, right? As vice principal also pointed out, I think that's more important. And failures are stepping stones to success. What's important is what you have learned after these rejections or failures to improve your work, and how you and your work have improved over time, right? And most importantly, be unapologetic to ask feedback, iterate, validate, and repeat, and never settle down until you have achieved what you set out to solve initially. These will eventually be the source of fuel for your scientific curiosity to drive the spirit of innovation. Ideas might work, ideas might fail, but what's permanent and compounding is your learning, experiences, and the skills. Focus on that and never compare yourselves with anybody else. You'll be on the right track. However, in this journey, you might experience pain, fatigue, despair. You want to give up, right? You might be scared of what people will think about me if things don't work out, and so on. Drawing from my personal and limited experiences as a biomedical engineer and a research scientist, there's one thing that I feel will help you in this journey, which is the power of belief and consistency. Believe in the world that you want to create and live in. Believe in the vision and mission you've set out to achieve for this. Believe in yourself, your journey, and the team and your pack that you take along in this path. Now, consistently repeat the same every single day. This power of belief, compounded by consistency in efforts, will ultimately increase your chance of success and help you to achieve what you set out for. I wish the very best for all of you today participating in this event, and I look forward to seeing and being a part of the world that you want to create. All the best and good luck to everyone. And thank you, Dr. Manisha and the BMSCE faculty for giving me this wonderful opportunity. It's an honor to be here today.
Thank you for your kind and encouraging words of knowledge and advice, sir. We're truly delighted to have you with us today. The principal could not join today, but he sends across his best wishes. Gathered here today are participating the innovative ideas and presentations to our judges today. Mrs. Sumati Mishra and Mr. Shubham Anand of Electronics at Neuron Technologies Private Limited, Bangalore. Her extensive expertise and experience in the field of medical electronics for many years shall surely be of incredible value for today's national project competition. We welcome you, ma'am. Our second judge, Mr. Shubham Anand, is the Associate Director of Research at Synaptic, an alternative data insights company. He has a work experience of many years, having worked in companies such as TrackXN and Oracle Corporation. We are honored to have both the judges grace the competition. I now request our first judge, Mrs. Sumati Mishra, ma'am, to share a few words with us.
Thank you, Vani. First, I would really like to thank BMS College HOD, Dr. Manisha. Thank you for inviting us to be a part in such a wonderful event. And I would really like to thank Dr. Vijayalakshmi also to having me on board. And as Dr. Shripat said, it's very true that biomedical engineering is one of the most innovative field in terms of engineering. You come up with idea, it requires a lot of consistency because you will be working on an idea which is novel, which is quite innovative. Nobody will be working on it. So most probably you will be starting it from scratch. And when it comes to engineering, working in the field of biomedical engineering is something in which you'll get a lot of practical experience. You will be working with hardware electronics. You will also use so many diverse field to give it a shape of something as a product, which will definitely impact the ecosystem around you. So it's one of the best and wonderful field to work in. You will get a lot of experience working in it, and if you have that instinct of you're working in the field of engineering, it gives you a lot of pleasure working in it with a lot of practical knowledge. So all the best to the teams who are going to participate, and great to be here. Thank you.
Thank you, ma'am, for those words of knowledge and encouragement. I now request our second judge, Mr. Shubham Anand, to share a few words with us.
Hi, everyone. A very good morning. Thanks a lot to the professor and HOD from my alma mater, Medical Electronics, BMS College of Engineering. I mean, a lot has been said and already, so I don't want to take a lot of time, but I just want to talk to all the participants directly. You all should be feel very privileged to find such an opportunity at this moment, and these things seemingly very simple and very straightforward at this moment, and then you never know if you keep doing this at one point of time you'll be doing, and then will be very deep in very high tech and innovative projects. So use this as a learning opportunity. Focus on your peer learning, what you learn from all other participants, what they are presenting. Try to network with them, figure out how things work, and all the best. That's all.
Thank you, sir, for those kind words and for being a part of the competition today. I would now like to request Sakshi to brief all the participants about the guidelines of the competition. Thank you.
I hope all the contestants are well prepared and ready for the competition. We hope all the contestants are through with the guidelines by now. If not, let me take the opportunity to brief you all with the basic guidelines of the competition. You'll be given 15 minutes to present your work. First 12 minutes, you'll be allowed to present the project, and next 3 minutes shall be for a question and answer session with the judges. During your time slot, it is mandatory for all team participants to be present, unmute themselves, and turn on their videos, followed by a quick introduction to begin with. You may not be present while the other teams are presenting. Please see to it that your audio and video connectivity are all in good condition for smooth operation of the competition. Before I conclude, I request you all to switch on your videos for a small group picture.
We once again request you all to please turn on videos.
With this, I would like to take the opportunity to deliver the formal vote of thanks for this short inauguration with immense pleasure. Firstly, I would like to extend a heartfelt gratitude to our beloved principal, Dr. S. Muralidhara, Vice Principal Admin, Dr. Suresh Ramaswamy Reddy, Vice Principal Academic, Dr. Bhim Shah, for gracing us with their presence today by taking out time of their busy schedules. I would also like to extend our gratitude to the chief guest for the day, Dr. Shripad Krishna Devalla, for being a part of this competition. A heartfelt gratitude to the head of the department, Medical Electronics Engineering, Dr. Joshi Manisha, and Dr. Vijay Lakshmi K. Ma'am, Professor of the Department of Medical Electronics and Engineering, and the judges, Mr. Shubham Anand and Mrs. Sumati Mishra, for gracing us with their presence today. We are extremely thankful to all the faculty members and all the contestants present for their patience throughout this inauguration. Thank you all.
Thank you.
Thank you all once again, and I wish all the best to all the contestants present here.
I request the first team, Team Atlanta, to begin their presentation. I request the first team, Team Atlanta, to begin their presentation, please. I request Sushmita, Monica, Saran, and Sriram, if they're present in the meeting, to begin their presentation. I request the second team, Team Medwingers, Sobika, Bharat, Vashini, Vishwa, and Danish to begin the presentation.
Good morning, ma'am.
Yes, you're audible. Please start.
Sorry, I'm facing some issues to present my slide. Can I take some more minutes?
I request the third team, Team Fantastic Five, M. Pavitra, Jinoline Pakya, Jashika, Sarmila, and Gopinath to begin their presentation.
Good morning, ma'am.
Good morning, you're audible.
Ma'am, we can't share the PPT, ma'am. It's showing that only the host can share the PDF, ma'am.
Please check again, you should be able to present now.
Yes, ma'am.
Good morning, one and all present here. I'm Jashika, and here are my teammates Pavitra, Jinolin, Gopinath, and Sharmila of Hindustan College of Engineering and Technology, Coimbatore, to present on a topic called low-cost pyrometer in this national project competition, Biomed Technophilia 1.0. Our spirometer is used for measuring volume of air we inspire and expire. It identifies two types of abnormal ventilation patterns and detects the abnormalities of various lung conditions in human respiratory system.
By which by sitting far away from the patient. the future of web connection can be integrated with many other medical devices for other diseases. And the methodology we have implemented here is to provide a pressure sensor, and we can provide the device with a pre-programmed setting for medical conditions so that the device can be used by people without an experience and any knowledge of use.
Components require hardware, PVC pipe, Arduino, pressure sensor, plastic tubing, ADC, LCD display, software embedded C. Block diagram. When a patient blow air into the tube, it passes to the pressure sensor and Arduino. And we have already encoded code in Arduino. So it converted into pass to the ADC. Then it display the value in LCD. This is a circuit diagram of our spirometer.
The main advantages of our system is it is easy to test and it is portable. That the project is very cost effective. It is handheld and durable. The disadvantages of our project is the patient must be prepared to do the test in advance. Sometimes due to the patient's illness, condition must be repeated the test any times.
Application, the device can be used to test functional lungs and respiratory system. Future scope, we can add additional LCD screen to use a touch screen in combination with the graphical user interface to make programming progress more initiative and to show the exact result on time and to submit with order that restart the device and to be ready for specific test. We can also use different microcontrollers such as Arduino Mega to use more input and to provide faster processing. Also, we can use artificial intelligence so that it can detect the problem and output the proper currents accordingly. It is also possible to implement a Bluetooth device so it can be controlled wirelessly.
Result. Our project was to build and test a proof of concept prototype of a low-cost pyrometer. We have concluded that our system is an acceptable proof of concept, although additional improvement and tests would be desirable. We have shown that such a system is feasible. We are confident in our calibration of the electronic sensor and security, but for full accuracy, the flow transducing device should also be calibrated with a known flow source to determine how the conversion from a flow rate to pressure difference varies from the theoretical equation.
In this project, we have configured and implemented a low-cost pyrometer with the differential pressure transducer being the heart of the device and tested its feasibility and reliability with a simple experimental setup. The software that we have created is not device-specific and is an executable file which can be used with any device. After multiple hardware tests and simultaneous software simulations of the networks, we propose this particular model of spirometer. Our proposed device is portable, compact, rugged, very economical and user-friendly. Here we have attached the references of our project. Thank you.
That was the presentation by Team 3. I request the judges to begin the Q&A session.
Hi, can you please explain that what you will be using, what will be the end product first? Like what result I'll be getting as a user?
Ma'am, we can measure the air volume of the lungs, ma'am.
Air volume of the lungs. What unit I'll be getting? Let's say a user is measuring his lungs volume. What will be the value which I'll be getting?
Pascal, ma'am.
Okay. Second thing, you have mentioned that you have to use an additional LCD. Is it possible that you can use only one LCD? It can work as a touch also, and it can show you the result also. Why you need an additional LCD?
Ma'am, we have used only one LCD, ma'am.
Yeah.
But if need in future, we can add it, ma'am. But we are not okay with the idea. We are not aware of that, ma'am.
No, you have mentioned that you will be using an additional LCD. My question is that only, can you name me one LCD which can be used, just one, which can, show you the function for touchscreen also, as well as it can display a result also.
Sorry, ma'am, no idea.
Okay. Okay. I guess that's it from my side. Thank you.
Thank you, ma'am.
Hi, Shubham here. I have a very basic question. We talked about spirometer, and yes, Mrs. Sumati talked about the unit that you would be using. So the team, the common question to all of you: what's the normal value uh, that we measure using a spirometer?
$120 or something, sir.
Okay. And what would be the sensitivity of this device? Would it be able to measure the difference if it is deviating?
Sorry, sir, not getting your question.
So that's the normal value. Let's assume if that's the normal value, what is the sensitivity? What delta or the change in the from the normal value would be measurable?
Maybe 20 or 25%, sir.
Okay. Got it. All right.
Yes, sir.
Okay, if anything is there, you must.
Thank you, Team 3, for the wonderful presentation. I would next like to call Team Healers, Shreya, Samskriti, Shreya, and Monica to begin the.
Am I audible? Hello?
Yeah, you're audible.
Yes.
Yeah, thank you. So good morning. We are Team Healers, comprising of myself, Monica, Shreya Patil, and Samskriti. We're from the Medical Electronics Department. And our topic for today is phonoelectroencephalogram. Next slide. Okay, so EEG is a test that detects abnormalities in the brain or electrical activity. So during the procedure, electrodes consisting of small metal discs within thin wires, they are pasted onto our scalp, from which brain signals are derived. Vision is the most important sense of human perception of space. So information about time dynamics relating to sound. Hence, we have come up with a phonoelectroencephalogram, in short, PEEG, which is the addition of sound to EEG waveforms. Next. So the main disadvantages in the present EEG is poor spatial resolution, visualization of minute variations in frequency ranges of different types of EEG, or not that evident, and lack of immediate validation and analysis. Our solution is transducing EEG waveform to different sound frequencies, which helps in better understanding of the waveform. Our goal is to make a highly efficient prototype that helps with neurodiagnosis. So basically, the aim of PEG device is to convert brain signals to sound waves, which makes the diagnosis easier and faster. These representations are expected to be useful for medical practitioners and neuroscientists.
Amma, I have to go for my project presentation. Please don't call me.
Okay, this is a block diagram. The electrodes are connected to the frontal lobe, and for the reference, in order to get the EEG waveform of the activity, the jackbox is used for the selection of the electrode placement. And then, since the acquired signal are very low, they need to be amplified and filtered. After the filtration, it is sent to the analog digital converter, and the output is printed. And the second part is sent to the frequency modulator, from which it is converted to sound. And this is the hardware circuit we have done. We have done the hardware circuit of instrumentation amplifier and notch filter. The output from the notch filter is fed to the Arduino board, and the data from the Arduino board is recorded and put into the MATLAB code for the extraction of EEG signal and converting the EEG signal to sound.
We have recorded the implementation, and this is the video. We have connected the electrodes to the frontal lobe and the reference over here, and then that signals are connected to the 3 instrumentation amplifier with a gain of 100 each, and totally summing up to 10 power 6, and that is given to the notch filter for 50 hertz noise cancellation, which is then connected to the Arduino Uno, so that the signals are connected to the computer. These signals are taken and the waves have been detected. That has been made into a DAT file and then given it as an input to the MATLAB, where the data from the Arduino is taken and it is given as input, and EEG waveforms are classified based on different frequencies. These are considered in order to convert them into different audios. These are the results that we got from the MATLAB code that we used to extract the different EEG waveforms. First one is the EEG, and then the other 5, as we know, 5 EEG waveforms, they have been classified. And these are the audio sounds that we have got as output.
This is the alpha.
Shreya, we can't hear.
Okay, wait.
No.
Could you hear now?
No. Can you join on phone and then on your mic, unmute yourself over here?
Yeah, wait one second.
Just sorry for this disturbance. These are the outputs that we got for 5 different waveforms of EEG.
The applications, while EEG technology has been around for the better part of the last century, it wasn't until fairly recently that it was made available to the general public. So we can use our prototype in various kind of fields like neuromarketing, where economics use EEG research to detect brain processes that drive consumer decisions, and in human factors, where we can see various in social interaction tools, and in clinical and psychiatric studies, and in brain computer interfaces. The scope of future work, the prevalence estimates in India for 2019 range from 3.0 to 11.9 per thousand of the population, and incidence from 0.2 to 0.6 per thousand of the population per year contribute to the number of people suffering from neurological diseases. Misdiagnosis is dreadful, and with the help of PEG, the data based on number of neurological disease patients and the number of misdiagnosed EEG cases can be reduced. The various applications, we automatic event detection, storage, quantification, and networking capabilities. It reduces the investment on unwanted treatment due to misdiagnosis, and we can access appropriate treatment for the required disease with a less chance of misdiagnosis. The disadvantages, the complex EEG waveforms cannot predict from where the EEG the EEG waveform is generated. And this our PEG can help in reducing the cases of misdiagnosis, but does not predict the origin of the waveform. Coming to our conclusion, looking at the data based on the number of, this is the pictures of our project. The first picture is the instrumentation amplifier, and the second picture also consists of the notch filter, and the third picture is the Arduino Uno board which we used. Coming to the conclusion, looking at the data based on number of neurological diseases, disease patients, and the number of misdiagnosed EEG cases, we have come up with this idea which might help in diagnosis, and we will be working on the output audio sounds we get, which will be much easier to predict the EEG waveform. Using our engineering skills, we plan on building a prototype and test it on different EEG reports of various patients being recorded. We are planning to work on generating different frequency sounds for common actions. Lastly, we will research on misdiagnosed EEG cases, the diseases, the errors, the reason behind the misdiagnosis, and improvise it further if needed and bring the product to the market. Next slide. These are the references we have used for our project. Thank you.
I request the judges to cross question team 5, the healers.
Hi team. Well, I have a very basic question on the similar lines itself. So what's the frequency that human can hear? What's the frequency range? Any idea? Because my follow-up question is essentially based on that. Because we heard some audio there. I'm curious to understand. I do not remember the exact numbers, but delta and theta, these waves are below what humans can hear, or else we would be hearing everyone's brain waves walking around. So what was I hearing? That's my simple question.
We have amplified the signals.
Yes. Because the signals are very small, we can't use it. By using frequency modulator, we increase the amplitude of the signals.
So we use frequency modulator to increase the amplitude.
No, we use amplifier to increase the amplitude and frequency modulator to convert it to sound.
Okay. Okay. Fair enough. All right.
Can you tell me what are the 5 frequency ranges you're taking? Because just by hearing it, we actually can't make any difference. Can you tell me any range you're taking? Am I audible?
Yes, ma'am. Ma'am, we have considered the standard frequency range, that is alpha, which is delta, which is lesser than 3 hertz, and all of that.
But if you imply it like practically, there be some variation or some range of frequencies. No, it cannot be just exact value. What's the exact frequency value for delta? If you converted it, there'll be range of some frequency, no, for all 5. Because as a user, I can't make out the difference in all 5 audio. So frequency will be a better example.
Ma'am, it is delta from 1 to 3 hertz, theta from 4 to 7 hertz, alpha from 8 to 12 hertz, beta from 13 to 30 hertz, and then gamma, which is greater than 30 hertz.
Nice. And second question is that do you have any control? You're taking, what's your control parameter by which you are detecting that the data which you're receiving is correct or not? Do you have any control data from which you're comparing it?
Ma'am, we are not comparing the data. Like we get the signals, and then we based on the waveforms, we are converting it along with the waveforms to sound. So as we all know, like audio adds on to the visual representation. Based on both, the disease can be diagnosed even more better than just the visual is what we came up with. So.
Yeah, but right now what data you have received, if there is no control parameter, it's how you're rectifying that what you're receiving is not just noise ratio. It's correct.
Mom, we're still working on it. Our first step was to convert the waveforms to this. We still have to work on each diseases for what is the standard value and how it will be different from other diseases. In case of epilepsy, how are the differences? This is the first step towards the project.
Yeah, we'll be taking those data and then working on that now. For now, we have considered this.
This is the first step of our project.
Third thing, do you have any plan if you want to, how your data will be communicated to a system, how the doctor will see it? Right now, as a microcontroller, you're using Arduino, right?
Yes.
Can your Arduino Uno simulate the data and send it to the system?
Yes, mom.
Uno can send it to connect it to your laptop and I can see the waveforms.
No, mom, you have to like code it and then using MATLAB, you can see that. Yeah,
but what will be the end result? How the user will see it? is there any plan, any connectivity you want to add, like any module for BLE or any module for AI, or how you're going to connect it?
Ma'am, the output is just the sound, so I don't think we'll be using any AI for now. It's like, yeah.
Okay, that's it. Thank you.
Thank you, Team 5. That was an excellent presentation. Next, I call upon Team 6, Team Ray, Arpita, Rakshit, Yogeshwar, and Amog.
Thank you. We are Team Ray, myself, Yogeshwar.
Good morning. I am.
Good morning. I'm Amog B.
I'm presenting my screen just to confirm.
Yes, it's visible. So, good morning, everyone. We, the team of Ray Health Tech, consisting of myself, Arpita Kumar, Rakshit K. Shetty, Amog B, and Yogeshwar S, present to you our mini project, gesture-based monitoring for partially paralyzed patients. Next slide. So ischemic stroke is one of the most common types of strokes that occur across the globe. And currently it's at 87% across the globe. And it is mainly caused due to the brain blockage or clots caused in the artery of the brain. So in the entire process, approximately 1.8 million people in India suffer from stroke every year. And it is only early treatment that can reduce morbidity and mortality. In the entire process, a patient can't be monitored continuously for any help or assistance, which can lead to many serious health problems in case of an emergency. Next slide. So the objective that we are trying to solve with this particular monitoring system is to ease geriatric care in household, ease communication in stroke patients, mainly because when a person is being affected by stroke, they basically get their speech totally off, and since they are partially paralyzed, only half part of their body goes out and only the other half works, which thereby affects either the arms or the legs and makes it difficult for communication. And finally, it is to help monitor the recovery rate of the affected patient as well. Now hand it over to Yogeshwar. Next slide.
Thank you, Arpita. So as the name suggests, it's gesture based. So initially we had to track the movements of the hand or the device where it is attached. So we'll be using a gyroscope for that. We could analyze the data from the gyroscope for detecting which type of gesture. For example, it is left side or right, up and top, up and bottom. That is what we have set for as of now. Then upload the data in suitable IoT storage or cloud storage. Here we have used Firebase. Firebase two major parts. One is Firebase real-time database, which will continuously monitor the sensor data. And another one is Firebase Firestore, which will collect all the user data like patient name, equivalent gesture commands, which are totally editable. And the data can be visible in our website, which is active also. And if there is any requirement, if the caretaker takes his hand up, then the alert will be gone to the caretaker or family member through our website. So this is the flow, how it works on. Data will be sent by gyroscope, send the data to cloud, that is Firebase here. That will be collected in our website alarms, and the stroke patient can be attended. So implementation, we have majorly used two major components. One is ESP32, that is Wi-Fi and microcontroller, and gyroscope, that is MPU 6050 to sense the gestures. And we publish it to IoT cloud, where that is Firebase here, and battery and charging circuit, and a small push button to just power it on and power it off. So as Gyroscope MPU 6050 follows I2C or inter-integrated circuit protocol, the connections were as followed. It follows SDA and SCL line and 3v3 to VCC and ground to ground. When we come back to the website part, it was built on React.js. It is a framework by Facebook, and not framework, it's a library more precisely. And for styling components, we have used Style1 CSS, which is available in net website. And it is used for tracking the information of the sensor and editing the information of the gesture and details of user. It also has login functionality. We have used Firebase email and password login authentication system to log in the user. The same details, email and password is fed to the microcontroller as well. So there'll be only that same user's data will be transferred within the website and the microcontroller. It is basically for security reasons, and no one else can access the data. And the Firebase real-time database helps to continuously monitor the sensor data. And Firestore helps us to store all the personal information. Thank you. Now I pass it on to Amog.
Good morning. My name is Amog. I'll be explaining about the disadvantages of the product. The battery part was the problem due to some constraints which can be done later using PCB, which consumes less power. In our case, we had 3.7 milliampere batteries, but the ESP32 wants a minimum of 5 volts, so we were not able to satisfy this. Even though we can connect the batteries in parallel and we can fulfill the 5 volts requirement, but the device would become bulky. So this was not addressed now, and the alert part was not able to address because there was no free software to work with. And now at present, the caretaker won't be receiving any notification on it. Next slide. The future scope, as discussed, IoT-based non-invasive health vital monitoring will play a major role in the field of healthcare. In this busy world, having a person dedicated to monitor a patient is hardly possible. Having said that, disadvantages and problems that physical monitoring process, it's always a great advantage in having a remote monitoring system. Hence, the device we have developed will have a great potential in the healthcare market. By using machine learning like algorithms, the device can be developed in an even more effective non-invasive monitoring systems. Also, by adopting complex programming, the device can be developed as a body vital measuring device and not just a monitoring system. Next slide. These are the outputs of our GSR sensor due to the gestures. If the person is moving his hand up, it was programmed that he wants water. And similarly, down, he wants to go to washroom. Left, I'm hungry. Right, want medicine. Next slide. This is the basic circuit diagram of the product. Next slide. Now Rakshit will be explaining further.
Yeah, thank you, Amog. So this is the picture of the website. The first page will be when we type in the web address, the first page that opens is the track patient page. And once we click on the track patient page, there will be a dialog box which will be opening asking the credentials to log in. And once we give the right credentials, the page will be logging in. And next slide, please. Yeah, so we get this, what is the gesture of the patient at that moment. And once we attend the patient, we can click on the attended button, and this will be stored on the database. Next slide. And here is the option to enter the details of the patient and the caretaker. We also can change the gesture, the message that each gesture will be giving in. For example, in this previous slide, the up button was drinking water. We can change it to something else here based on our requirement on our comfort level. Next slide. So the conclusion of the project was the device was around 99.9% effective with whatever electronics and circuit that we had implemented, and the signals, the alarms were given in time with a time lapse of around 3 to 5 seconds. And yeah, this project gave us a wonderful exposure to explore the teamwork, writing a technical paper, writing reports in IEEE format, web designing, and of course wide knowledge on electronics. And the future scope also is also has a lot of future scope, because implementing it in a large scale in the potential market of healthcare will be of a great advantage. Thank you. If there's any question, we would be glad to answer.
Hi team.
Hello.
So how would you be tackling the false positive? Let's say if no action was required, but somehow if it picks up, so do you just mark it as addressed or attended, or is there a way you capture that information as well?
Sir, it is the caretaker's way if you want it to put it as attended or not. It is not compulsory. We have put it to attended because to just record what and all gestures, or like for example, medicines are taken at the particular time. To just record those things, we have added it that. Yeah, false. Sometimes false will be there because that should be like sudden movements can be later algorithm. Using some basic algorithms, we can find out whether it is a false or a required gesture.
Okay. And then you guys talked about there was lack of any free software for notification or alerting. So
Yes,
what
sir.
all options did you explore, or did you come across anything which was premium?
Sir, actually, we have used Firebase. So Firebase itself has its something called as Firebase functions, which is actually a paid software. Paid software in the means we have to pay some basic premium over there to use that. It is easy to implement if we have Firebase functions wherein as soon as it detects a change in the real-time database for a particular gesture, then it will be automatically alerted using its own FSM, that is Firebase application messaging system.
Okay. So what
I
all
don't
would
think
be
so.
the
It's
channel? What all channels would be getting the alert? Is
sir,
channel.
I didn't get you.
So would it be mobile, website, or is it a simple SMS?
It's a mobile, which, okay, go ahead, go ahead. Okay, basically we'll be having the web page part of it, which will be alerting the person about if the patient needs help or not. So it'll be like an alarm, and you'll be getting it as a message. And once the patient is taken care of by the caretaker, they can just confirm it.
So essentially a web notification.
Yes.
Sir, it depends. We can. There are many possibilities. However, Firebase messages works. One can be a web notification, either through mobile phones. We can send in SMS as well, but that will be an extra cost, I believe.
Absolutely, absolutely. So the feedback here is that think about the happy flow. If the system operates, what would be the logical flow of this operation? Because the implicit expectation here is that someone should be literally sitting and staring at a dashboard and waiting for a notification, right? So you have to factor in those aspects as well when you are designing a system. What's the most appropriate way to send out the notification? That's the feedback. Yeah, thank you.
Yes, sir. Thank you.
Thank you, sir.
Thank you, sir.
So my question is, Amog was saying that the design was looking bulky. Can I ask you, the product will be in the form of wearable,
Yes,
or
ma'am. It's a wearable device.
wearable type of what? It will be a glove. It will be a
It's a watch type, ma'am.
Okay. Do you have any idea that you can make it in the form of gloves also?
I mean, we can include the gloves also, but in many cases, the stroke patient will having a bent arm. It's difficult for them to wear it.
Okay.
So some of their fingers end up to be bent, which makes it difficult for us to fit in each finger or each part of the glove to the finger. So which is why we have made it as a watch. So that itself will be tracking the movement, like
Okay.
up, down, left.
Okay. And second thing, that what's the voltage input you need right now? You said 5 volts are way too much.
As of now, we are using ESP32, which has a minimum voltage requirement of 5 volts.
Okay.
So
You can
if we bring it down to a PCB, we can use less voltage also, ma'am.
no. What's the current? That's what I'm asking. What's the current input voltage you need to run your circuit?
It's 3.3 volt as of now. So the minimum requirement is 5 volts.
Minimum requirement is of 5 volts. Then your ESP32 will work out in that case.
Yes, we are not able to produce 5 volts because of the bulky battery size. So as of now, with small battery size, we have produced 3.3 volts.
Okay, which battery exactly you're using? Can you tell me?
LiPo 3.7.
Okay.
Ma'am, to summarize it, I will give you a clear picture of it. ESP32 requires a bit more higher voltage when to connect to a Wi-Fi. Around more than the 5 volt or the not exactly the voltage, but it requires more power, that is ampere. It requires more current to connect initially to a Wi-Fi. So a LiPo battery, which is of 3.7 volt, is normally can give only around 0.2 to 0.4 amperes. So that is not sufficient for the initial connection of connecting to a Wi-Fi router or a network. So that is one major problem faced, and continuous data collection cannot be done within that amperes. So we need some higher amperes coupled with extra voltage to get more life, and we can connect it.
Okay, so here one feedback for you. You can also explore Seedunos.
Okay.
They already have inbuilt, and including your ESP32 also, you will find that ESP32 already have inbuilt neural networking libraries. So it will be easy for you if you want a continuous monitoring, real-time monitoring, and it will be easy for you to connect it through cloud.
Sure, ma'am.
And you can, if you can switch to Seeduno, your bulky design can also be in the form of compact wearable form.
Yes,
sure, ma'am. We will do it.
Okay, we'll do that.
Thank you.
Thank you, ma'am.
Thank you.
Thank you, Team 6. That was an amazing presentation. Next, we have Team 8, Smart Shoes. I request Kinnari to begin their presentation.
Yes, ma'am. Ma'am, it is visible.
Your screen is
Hello.
not visible yet.
I'm able to upload. Ma'am? Hello?
Yes, hello. Are you able to share your screen?
No, ma'am, I can't able to see.
Okay,
Can
would
you
you like to
provide
go
me some
next?
time?
Shall we call upon the next team and you'd like to go next?
Okay, ma'am.
So next we have team 12, Maya, Mantra, Tarani.
Hello.
Hello, yes, you're audible.
Hello.
Hello, you're audible. Are you ready, Tarani?
Hello. Very
Hello.
good morning, ma'am. Actually, I'm in ICU. Some network issue is there, so I can't able to connect with you.
Okay.
I'm trying actually. Can I come back after 10 or 15 minutes, ma'am? Can you give 15 minutes time?
Yeah, that's all right. So we'll have the next team. Next team is team number 3, Medwingers.
Ma'am, can we send our PPT? Shall we present this?
You may share your screen now.
Okay, now.
Are you able to view the screen? You may start your presentation now.
Yes, ma'am. Thank you. Hi, everyone. This is Sobika from Hindustan College of Engineering, Tamil Nadu, Coimbatore. Our team members' name is Sobika, Varshni, Bharat, Vishwa, and Danish. We are from Adventures Group. Our product guide is Miss M. Deeparani. Then we are presenting the pill reminder system. Next slide, please. Basically, what is nowadays the technologies are updating with many development. We are forgetting to take our medications at the correct time. So we are decided to give an alert to take medicines. Various diseases in human bodies, in taking of food or other technological environment. Severe condition to be focused and help for aging patients. Then they are loss of memory to gain benefits of clinical prescribing drugs. Next slide, please. What is an objective of this reminder system is at the time of the medication system, many errors are taking place due to the pill system. Then we are introducing a smart pill type of system because we can able to take medications at the correct time by the using of notifications. Next slide. By the hardware system, the proposed solution is most of the older people are forgetting to take their medications, or tumor patients or such as any can be useful at this project. Then the project is help the older people and people with disability. This is not a big equipment. It can be made at low cost. It works based on the current and its maximum current capacity at least at a 5 volt. Next slide, please. We are using some software equipments like embedded C and Arduino IDE compiler. We are inserting some programming to the this kind of requirements. Then we are using some hardware requirements also. Then Atmega microcontroller with some power supply varying from 5 voltage. Then we are using APR voice model for the voice. We are giving some input to the voice, whether what is the tablet is to be. Then there is in the LCD display, the tablet name was displayed. Then we are using IR sensor. According to the number of tablets, we can use the sensor. Then for the reminder purpose, we are using buzzer. Next slide. This page was followed by my friend Varshni.
This is the flow chart of smart pill reminder, where it starts and when it is high, the pill will be available. When it is low, there will be no pill available. And when there is no pill available, it will tell us the empty voice from APR, and it's as a time reminder, and when give us a voice recognition from APR. Next slide. This is the block diagram. Arduino, the display screen, voice module, speaker, buzzer, power supply, and two IR sensor are used in this smart pill reminder. Next slide. And this is the same circuit, which can Arduino, UNO, voice modulation, LCD, and two IR sensors, buzzers. Next slide. Result will be tell by versioning.
A result of our project is to attain independence in taking of medicine to our body in correct time, and correct optional medicine should be important. And many people can manage it, but it is difficult to some people to take medicines in correct time, especially older peoples. It is also told that especially designed for older peoples, for that the right reminder equipment has to be designed based on the severity and type of disability. And future biomedical engineers, it is our duty to develop newer technologies to assist patients who has this type of memory disability. Next slide, please. And advantages of our project is it is portable and it is easy to use and it is a low cost. Disadvantages of our project is signal may low sometime and low balance in SIM can't be found. Next slide, please. And future scope of our project is reducing documents by introducing OCR, and the mobile application can be updated with OCR systems, and it can be trained to precisely understand a doctor's handwriting over time and after scanning any doctor's handwritten prescription or report. And cloud-based multi-end application. It can be developed and made cloud-based, and doctor versions of the application can be developed so that the doctor can access their patient's data easily. Next slide, please. Security reasons, and we can secure systems so that dosage information cannot be changed by unauthorized users. And real-time video, it can be using real-time video processing by which a caretaker can see a video of the patient concerned by being away from patient. And it can be involved with all the hospitals, clinics, or any health centers. Next slide, please. And limitation, elderly anonymous pill dispensable people find it difficult to flip the device and less likely to use Android phone. Smartphones can be less used by elderly people, and blind people cannot use this, and it need to press the button on dispensing unit. And PA camera gives error sometimes and need to reboot the processor. Next slide, please. Conclusion. And to attain independence and taking of medicine to our body in correct time, and correct time especially older people's, it also special design for older people's and memory disability. Next slide, please. And it is a reference of our project, and thank you.
Hello guys. So my first question is, why specifically you're using IR sensor? What's the use case of IR sensor?
Ma'am, in order to sense the tablets, we are using that, ma'am. By the reflection of the light, it can sense us, ma'am.
Okay, what's the wavelength you're using then? Because if it's IR, it's you cannot see the color from your visible range. The human cannot see it. That's why you're using IR. If IR,
Yes,
then what's
ma'am.
the range you're taking for detection of the pills?
It's up
What's
to
the
6
range?
to 700, ma'am.
What? Can you please repeat?
600 to 700, ma'am.
What's the, is it IR or is it near IR? NIR or it's IR?
Ma'am, I can't get it, ma'am.
There are in the range of infrared, there is one which is near infrared, there is one which is infrared. So your measurement will come in NIR or it will come under IR?
It come under an IR, ma'am.
Okay. If it's coming from NIR, your range is starting from 600 to 700. What if the pill is of some purple color? Then the range will be more than 700.
Yes, ma'am. According to the pill, the IR sensor we are adding much, ma'am.
But if it's
we are
just
giving
color,
some distance between the box. Yes, ma'am.
What's the distance?
We are under the pack cover will be the pills are into the box, ma'am.
Okay. The second question is why IR? If it's just color detection, you can use the calorimetric sensors also, no?
Yeah, yes, ma'am. First we are giving some prototype. So according to that, we can change it, ma'am, according to color.
That's the feedback, dear, that you can use the, if it just based on color difference, it would be better if you use which can differentiate between color, not in terms of wavelength.
Okay, ma'am. We will implement next time.
Okay, and is it a monitoring system? So let's say you're talking about the elderly people. So let's say they forget this pill box in house and they're somewhere out. Then how they will get notification?
Ma'am, it will connect to some mother, father, or son also, ma'am. By using online or offline, it will remind to us, ma'am, to take the tablets. We are minimizing the size of the tablet box so they can take.
But the size of your pill box will be completely proportional to the size and the quantity of your pills also, right?
Yes, ma'am. It's based on the number of pills.
Okay, but then how many data you are planning to feed to your system so that it can rectify and differentiate between the pills? Because there are so many, the pills which are available. And let's say if some patient is getting a generic medicine, which the component of the medicine is same, but the color is not same, then the device will not work in that case.
Yes, ma'am, but we can implement more sensors according to that, ma'am.
Okay. Thank you.
Thank you, ma'am.
Hi, team. So, when you assemble the entire system, what's the cost of the prototype?
Sir, it will comes under 1,000 to 1,500.
Okay, and what's the most expensive component of this entire prototype?
Arduino board, sir.
Okay, okay, fair enough, fair enough. That's all.
Thank you, Team 3, for that amazing presentation. We next have Team 14, Roshni A. Ramesh.
I'll be sharing my screen. Is my screen visible?
Yes, Roshni, your screen is visible. You can go ahead.
Okay. Okay, so good morning to all the judges and one and all present here. I'm Roshni Rameshan, 4th SEM, final year MTech student from BMS College of Engineering. So today I'll be going to do my project presentation here, which is titled as Bone Cancer Detection Using X-ray Images under the guidance of Dr. Joshi Manisha S. So these are the contents which I'll be dealing today. So coming to the problem statement, any type of cancer it may be, there are lots of procedures to detect what cancer it is, which may consume money and time. And there can be also misdiagnosis happening where the radiologist might find it difficult to interpret the medical images. In order to overcome all these things, have built a clinical assistive tool. So this tool basically consists of a cascaded deep learning model where it will be able to classify whether the given image is a cancer image or a normal image. And then if it is predicted as a abnormal image, that is a cancer image, the segmentation will be done where the cancer portion will be segmented, and depending upon that, stages will be predicted. So the software which is built over here is a user-friendly software. So while talking about the cancer, here I'm concentrating only on one type of cancer, that is bone cancer, and under bone cancer, a type of cancer called as osteosarcoma cancer. So coming to the methodology, so this is the basic methodology. So first is data curation. Data curation is nothing but collection of all the data. So here, the data collected by me is X-ray images. So it consists of both normal X-rays as well as X-rays having the cancer, that is osteosarcoma. Then next is data storage. Data storage is nothing but you're storing the data. Next is data sorting. So basically, whatever images we have collected, those images won't be labeled. They won't be labeled. So it is essential to label these images. That is nothing but data sorting. You're basically categorizing the image. You're putting it into different categories. So here categories are nothing but one is the healthy bone and the other one is the cancer bone, that is osteosarcoma. Next comes data labeling, which I'll be dealing in the next slide. Similarly, data augmentation. Then comes the AI modeling. So here basically three models are running. So one is classification model where it basically classifies the given image is cancer or not. Then segmentation. So once it is, as I told, once it is classified it as an osteosarcoma, the tumor portion will be segmented. Then depending upon the measurements, the stages will be detected. So as I told, this is the labeling. So data labeling is nothing but you're basically creating manual mask. So here, this is done using Label Me platform, where you give the X-ray image, and using a tool called as polygon tool, we are basically putting boundaries around the tumor region and the healthy bone region. So the mask is predicted. That is the healthy bone region is nothing but the green, which is shown over here, and the red region is the tumor portion. Data augmentation is like actually, as the data collected, it's of a less in number, so we have gone for data augmentation. So because it's good if you give the model a good number of data. So we have basically used over here 90-degree rotate, horizontal flip and all to do data augmentation. Then comes the cascaded AI model. So this is the block diagram of the cascaded AI model, where you're basically giving the X-ray image, and then the X-ray image is first going to the classification model. So classification model over here is done using AlexNet architecture. So the model predicts whether it is a normal image or an osteosarcoma image. Once it predicts as an osteosarcoma image, it then enters into a segmentation model. So segmentation model, here the architecture used is ResNet architecture. Once the segmentation model predicts the segmented area, then staging algorithm is applied on it, where the stage prediction is done. So this is the staging algorithm. As I told, from the segmentation model, you will get the tumor portion. That is the mask basically. So you're calculating the dimensions. Then depending upon the standard X-ray, with respect to that, you're resizing the image. Applying some morphological operations, you're smoothening the edges of the tumor portion, and then smoothening the entire image, finding the contours, finding the area, and then you're finding the longest longitude and latitude of that, and then finding the distance. With the depending upon the distance, you basically estimate at what stage the cancer is. Coming to the implementation, as I have told, for classification model, I have used ResNet architecture. So this is the graph which I have got for the ResNet architecture. I mean, epochs versus the loss, and this is the confusion matrix. The segmentation model, as I told, I've used ResNet architecture, and the first set of images are the target and the predictions made. So as you can see, the predictions made are almost similar, almost accurate. Coming to the results, classification model. So basically, I have not only done experiment on AlexNet, but also conducted experiments on two other architectures, that is ResNet and VGGNet. So out of this, I have found that AlexNet gave a better performance. So the confusion matrix and the accuracy of the models are given here. And then similarly, even for segmentation model, AlexNet and ResNet were the other two architectures used. So out of those two, we have come to a conclusion that ResNet gave a better performance, where you could get smoothened edges of the tumor portion. So these are the ground truth and the predictions which are made. Coming to the cascaded result, this is the normal case. So here the image given is a normal image. So as you can see, it is predicted, the model predicts that the given image is a normal image, and the model is 100% confident in that. This is a case of osteosarcoma. The image is a normal image which is given. This is the original image. And this is the prediction which is made, that it is predicted it as an osteosarcoma case. So once it is predicted it as an osteosarcoma case, the segmentation is performed, where as you can see, the green indicates the healthy region and the red indicates the tumor portion. These are few more examples of osteosarcoma cases where you can see the prediction that it is osteosarcoma is made, and the segmentation done on that. Coming to the application. So here basically we are able to distinguish the healthy region as well as the cancer region accurately. Then one more thing is, we can also detect the stages of a cancer within an early stage, once you get the tumor portion correctly. Then this can also be used as a training platform, where the practitioners can train themselves in identifying the tumor portion and the X-ray images. And basically this is a clinical assistive tool. So apart from this, in the tool we are also giving other features, like where you can zoom the image, you can increase the contrast and brightness, blur the image, and also if the doctors are not satisfied with the predictions made by the AI, they can manually do the predictions and then find it out, and then detect the stages as well. So future scope is, as I have told, I have concentrated on only one type of cancer, that is osteosarcoma. So this can be built on various other cancers also. So there are various other bone cancers. So in future, different types of cancers can also be, class models can also be built. And here the staging model built is not that accurate. So we can also improve the staging model here. And in this platform, we can also include report generation, where automatically once the analysis is done, the reports can be generated. The disadvantage of this is, as of now, it's that, as I have told, the prediction which was made by the staging model is not that accurate. So this is one of the disadvantage of this. As I have told, this is the clinical assistive tool. So first is basically the doctor logins to the software, and then he gets his list of patients. So once he clicks onto a particular patient, this is it lands on this particular page where you have the PIDs of the patient and other features like, they can take different types of views like AP view, LV view and all. And this is the AI automated button over here, where once you click on the AI activated assisted button, the segmentation, the predictions will be automatically made. So yeah, once this, as I told, once this button is activated, this is the prediction which is made. So here it is written that the image osteosarcoma, it is an osteosarcoma image, and the model is 97% confident in that. And the stage over here is predicted as 1B or 2B. So this is the image, and this is the prediction made where the green indicates the normal and the red indicates the cancer region. And this is the and where the only the tumor portion is taken, and the measurement of the tumor area is done. I mean, tumor portion is done, and the stage prediction is made. So coming to the conclusion, as I have told, the stages can be detected at an early stage, and as I've told you, a software has been successfully built where we can detect osteosarcoma and the healthy bone, and the cascaded model performs successfully in detecting this, and even the staging algorithm is working fine up to an extent. These are a few references. Thank you.
Hi, Roshni.
Hi, sir.
Well, so I have a very basic questions. So you must have trained this model, right? So
Yes.
was that model available already to start with?
No, the model, this was not built. Actually, I built the model. Yeah.
Okay. So what was the sample size or of the training?
So basically, the data set which I have got was very less because osteosarcoma was not really available in a large number. So the total number of image which I have collected was around 231, like that. So in that, I have given 80% for training and 20% for testing.
Okay. And all 200 had that symptoms, right?
No, all the 200 didn't have the things. Actually, it was like there were 91 cases which were healthy, and the rest were osteosarcoma. So I included the healthy as well as the osteosarcoma so that the model could identify, it can differentiate between the both.
Got it. And what was the file format?
It's actually this one. Sorry, so JSON format. It's in JSON format.
Okay, in the JSON format itself.
Yeah.
And how is system able to ingest that? It's the JSON format for the image file. Are you sure about it?
Sorry, I didn't get you. So, no, whatever I have done, like the label me, all those things are in the JSON format.
Okay, so the labeling has been
yes.
Got it. And let's say again, it's a more of an exploratory question. A lot of these X-rays are not in digital format. So how do you take care of it, right?
As of now, yeah, I didn't. It was just the X-ray images. Whatever I have collected, it's a normal JPG one.
Okay.
So, yeah.
And then for the staging part, you mentioned that accuracy is not that great. So can you quantify that?
Yeah, actually, well, coming to the staging, it's not only with the X-ray, we can't determine at what stage the cancer is. So there are various other things done, like biopsy has to be, that's the main thing. So here, what I have done is only considering the measurements of the area, basically. So according to mine, it's like above 8 centimeter. If the cancer region, if you take the superior to inferior, if it's above 8 centimeter, then it goes under 1A or 1B. I mean, 1A or 1B. Similarly, if it's less than 8 centimeter, it comes into 1A and 1A, 2A, and if it's above 8 centimeter, it comes into 1B, 2B. So that this is one of the factors. Actually, the measurements is one of the factors. Other factors like the grade and the metamerisms and all, we can't identify in the X-ray.
But shouldn't these measurements be normalized rather than keeping it an absolute? Because individual body parts of size would be different.
No, this 8 centimeter is actually, it is done by the American Joint Committee. They have given this. So according to that only I have proceeded.
Okay, got it. Fair enough. Thank you.
Okay, thank you.
Hi, Roshni.
Hi,
I guess Shivam has covered all the technical questions. So I just have one small query. Have you tested what's the specificity for your app right now?
You mean for the architectures, what I have done for the classification, individually it was done. But overall, yeah, it is not yet done. It's still in the initial stages, just going.
Okay. Okay. So any chance yet that you can detect any false data?
Yes, as of now, it's detecting false data. The normal, some of the normal images, it's very less actually. Like I think 2 to 3 or something, it's detecting it as osteosarcoma, the normal healthy ones. So yeah, there are errors also.
Okay, okay. Yes, that's it from my side. Thank you.
Thank you. Thank you.
Thank you, Roshni. That was an amazing presentation. Next, we have Team 15 Bio Trio, Aditi Prasad, Vaishnavi, and Shruti.
Yeah, I'll be just sharing my presentation. Oh God. Shruti, can you share the presentation?
Yes. Just give me a minute. Can you see my screen?
Yeah, it's visible.
Is my screen visible?
Yeah, it's visible.
Can we start?
Yeah, you can start.
Good morning to all of present here. I'm Shruti, and my teammates are Aditi Prasad and Vaishnavi. Is my screen moving?
No.
Yes, it's visible.
Yeah, your screen is visible.
Okay. So our topic is the development of a biosensor for early detection of cervical cancer. So we are from the Department of Biotechnology, BMS College of Engineering. So cervical cancer is one of the most deadliest cancer, and it's around 2 in the number of deaths across the world. So these are one of the most used tests, which is pap smear, HPV testing, and colposcopy. So pap smear test is not a gold standard test, as the cancer is detected at the later stages. And when it comes to HPV testing, HPV may or may not lead to cancer.
And a colposcopy is basically done by viewing the cervix. So the cancer may not be seen if it's at the early stages, and it can lead to the observer's mistakes. So that is a point there. And so the first step in our designing of the sensor was the fabrication. And for that, we selected a candidate biomarker. The sample that we used is the CVF, which is the cervicovaginal fluid.
Shruti,
So as you can see in this picture,
the slides are not
slides are
moving.
not moving.
Yeah, manually select it.
Is it moving now?
No. Yeah, now
Okay,
it's
now?
moving. Yeah, no, yeah.
So as you can see in this picture, this is from one of the papers. They had considered 16 healthy and cancerous patients, and the concentration of our biomarker is specified. So above 20 was for the cancer patients, and below was for the healthy patients. Now, coming to the biomarker selection criteria, these were the 5 points that we kept in mind while selecting the biomarker. One is it should be a cellular-based biomarker rather than a HPV-based biomarker, so that it covers all the factors that could lead to cancer. Then secondly, it should be upregulated during the detection, so it's easier for detection. And third, it should be easy to work in labs. And fourthly, it should be detected at the precancerous stage. And fifthly, it should be very specific to cervical cancer itself. So we had a lot of queries. So we consulted various doctors, and and one of our query was like from any secretion, like how we know menstrual blood comes in and all that. Will that interfere with the detection of cervical cancer? And they suggested that it will not, because it's an organ cancer, it would stay right there. Now, coming to the objectives, we have 4 major objectives in this study. So firstly, we want to identify, and we have identified a cellular-based biomarker for early detection of cervical cancer. And secondly, we have fabricated a biosensor, which is very specific to cervical cancer itself using CVF samples. Thirdly, we want to test and optimize with the sensitivity of the biosensor. And fourthly, we'll be publishing the outcomes of this study. We have reached the second stage. We require more samples for testing, which we have applied for ethical clearance in HCG. Now coming to the workflow, so we collected 5 samples from Bhagwan Mahavir Jain Hospital, and we did ELISA based on that. And then we also started with biosensor fabrication, which involved polymer nanoparticle preparation. Then we coated a crosslinker on that, on the board. And then after that, we coated the antibodies. We also did dot ELISA so that we could understand the binding of the biomarker and the antibody and the sample, so that we can see how it happens in the surface of the biomarker. Then we also and the next step is the optimization of the antibody that we are using. Then we'd be replacing that with the candidate biomarker antibody, then testing with larger size, which we have applied for ethical clearance, and finally we'll be optimizing that.
So coming to the results, the first one was the validation of a biomarker, and for that we performed ELISA. 5 patient samples were obtained from the hospital with different ages, and condition history is seen here. So what we found out was the patient 4, who had precancerous cervical cancer, showed the concentration of 142 picogram per ml, followed by the P2, P3, P1, and P5. So since the concentration was very high in all this, we further went on to perform the Bradford's test. And from that, we could make out that P4, which was of our interest, had the highest biomarker concentration. Whereas coming to the P1, also it was a stage 3 ovarian cancer. But as we had consulted the doctors, we could rule out that there was no interference with the cervical and the ovarian cancer. So P5, which was endocervical and the endometrial growth,
they had a good concentration of biomarker, but then since again there is this overgrowth of the tissues happening, there is no interference with it. Next. Shruti, it's not moving. Yeah. So for the biosensor fabrication, every step we checked for the resistance. And so here we found out that polymer plus crosslinker, which was 2%, showed 75 kilo ohms, followed by 73, 75. And again, when the polymer and crosslinker, to that when we added antibodies, it was 85 kilo ohms, 84 and 90.8 kilo ohms. So it was like 3% showed a good change. And again, after adding the blood serum, it showed 2.2 kilo ohms. So there was a good interaction with the antibody and the crosslinker and the blood sample. So when we performed some analysis for the crosslinker concentration with 2%, 2.5 and 3%, 3% showed very good results. It was like a flower shaped structure was observed with uniform coating, unlike the previous two concentrations. So the next slide. So this is a clear image of 3%. And for 3.5% also, we did some analysis. It did give us a good analysis, good results. But since 3.5% is a higher concentration of our crosslinker and it can be cancerous, we have restricted ourselves to 3%. Next. So again, this was done with crosslinker along with the antibody coating. So antibody crosslinker concentration was differed, but antibody was not done. So 3% concentration again showed us a good results when it was combined with the crosslinker and the polymer. Yeah.
Coming to the FTI analysis, which was done to understand the interaction between the polymer and the crosslinker, the first 4 graph is polymer, which is doped with acid, and crosslinker, and the fifth one is just a polymer. So as you can see, there is a characteristic peak at the 3,429, which is seen in all 4 of the cross-linked one when compared to the 51. Next slide. Next slide. So the first table, it shows the vibrational assignments of the polymer, characteristic peak of the polymer, and second one is with respect to the crosslinker, which is found in 4 graphs, but not in the fifth one. Next. So coming to the FDI XRD analysis, so it helps us to understanding crystalline structure, which in turn will help us understanding the conductivity, which is a very important aspect with respect to the biosensor. So the green graph, it represents the polymer, which is doped with 2.5% crosslinker and 3% crosslinker is red one, and blue one is just the polymer. So now we consider 2 factors, which is crystalline size, which is D. So more the crystalline structure, more is the surface area, and hence more is the conductivity. So when compared with 2.5 and 3% add very good conductivity. And also the interchange separation length, it is inversely proportional to the conductivity. So 3%, it showed the overall higher conductivity when compared to 2.5%. Next. So advantages in a project is that since a sample is a cervicovaginal fluid sample, so the sample collection process is minimally invasive, and also it provides an opportunity for self-sampling. So there have been a lot of studies which has done that women are not coming up to take up the pap smear colposcopy, but this project gives this biosensor could help in giving them an opportunity for self-sampling. And also, as in the case of pap smear and colposcopy, there are chances that there are errors in the interpretation of the results. Since the biosensor is dependent on the resistance measurement, that could be voided, and it will have high specificity towards the cervical cancer, unlike HPV testing, which may or may not lead to the cancer. So, and also it could reduce the false positive. The duration of the detection would be lower when compared to the PCR-based and cytology-based testing, which typically ranges from 24 hours to a week when it comes to cytology. And skilled labors, the requirement could be decreased, and early detection, which is the main aim of the project. Next.
So the only limitation that we could get is with respect to since the entire detection depends on the antibody protein interaction, the incubation time is needed for that, which is typically 90 minutes. That is the limitation that we have for it. Next. So application, as said, early detection and specificity towards the cervical cancer. However, we have to validate our biomarker more and also test with more number of samples on the biosensor. So we have got the samples currently from Bhagwan Mahavir Jain Hospital, but we have cleared the ethical clearance at HCG to get more samples. And a provisional patent has been applied since we have a novelty with respect to our process. So this is the pictures of the project. Next. Conclusion is again early detection, eye specificity towards cervical cancer, and also since this is a chemiresistive type of biosensor, the operation is easier and downsizing possibilities are high. Next. These are the references. Thank you. We'd like to thank Department of Biotechnology and Medical Electronics of BMSCE, HCG, and even KSES. Yeah, that's it.
Hello, everyone. So the first question here is, I just want one clarity. So you will be coating your antibody with your biomarker?
Yes, ma'am. On top of the cross-linker layer, we'll be coating the antibody. And once the antibody is coated, we'll usually block it with BSA and we'll be washing it. And we can store it at the 4 degrees Celsius. So when the sample is obtained, we can just add the sample to it so that there will be a particular protein will interact with the antibody.
Do you have any docking data to prove your point that once you coat it with your antibody, it will show some affinity?
Ma'am, we did dot ELISA.
Yep.
So we did dot ELISA. So we coated the antibody, and then when the sample was put,
your voice is breaking.
Actions.
Ma'am, so when the sample was
got a brown dots with color change.
Shruti, you've muted yourself. So I'll answer the question, ma'am. So for the dot ELISA was basically done to know the interaction of how the antibody and the biomarker interacts. So for that, we got a color change, a brown color. So since the brown color cannot be depicted in the sensor, we have got a resistance change over here. So that will prove that there is an interaction going on in the biosensor.
So basically, it's a inline sensing that you're adding some cathode and it will record your impedance. How you're calculating your resistance? What do you mean by resistance?
Ma'am, what we usually do is that since our substrate is a PCB board, so we'll connect it with the crocodile clips and it will be connected to the heat meter. So what we'll do is that we have apply a very low current, that is 1 milliampere. So we get the voltage change to it. So depending on that, using the basic formula, we calculate the resistance.
Okay. No, that's my question. Then in later version, how you're planning to calculate the impedance? And why not color change?
Since the polymer, it is black in color, so the very mild red dot would not be observed on it, but we can observe it on the
nitrocellulose paper.
Okay, you'll be using nitrocellulose membrane to detect it. Yeah. So it will be in the form of LFA, or it will be in the form of some diagnostic device?
It will be in the form of a diagnostic device.
Diagnostic device.
So
nitrocellulose membrane is not incorporated into the biosensor. It's in the dot ELISA, but ours is PCB.
So for your product, for your biosensor, you will be using the nitrocellulose membrane, right?
Biosensor? No, ma'am.
No, ma'am. It's not being incorporated in.
Polymer.
Sorry, ma.
How will you test using your
polymer
nitrocellulose
based.
membrane?
Ma'am, nitrocellulose membrane is it's completely biology side. It is towards the dot ELISA. What we are doing in a biosensor is PCB board. On top of it, there will be polymer nanoparticle. On top of it, crosslinker. Since polymer nanoparticles cannot directly bind to the Fc region of the antibody, this crosslinker will act as a bridge between the polymer and the antibody. So on top of the antibody, whenever the particular protein, the interaction is very specific when we say with respect to the antibody and protein. Only certain let's say a particular IgG antibody can only bind to the IgG protein. So it's specific in nature, but we can confirm it using dot ELISA.
Okay, so small suggestion. In your product in later version, you can actually use nitrocellulose membrane and using the color change. Why I'm telling you this, the moment you are calculating the impedance or the resistance, once you will get in terms of some liquid sample, when you pour it, it will flow. Basically, you will get a lot of signal to noise ratio. So it's better to work on color change. And the best example to do it with nitrocellulose membrane. You'll get very, very fine data in that.
Sure, ma'am. We'll work on that also.
Nice. Thank you.
Thank you, ma'am.
Thank you.
Well, I have a hi team. So I have a very basic question. I just want to go well in the beginning of the slide. My question is about the choice of biomarker. So I think you did mention a bit around the ease of access of a certain kind of biomarker. I guess HPV is slightly invasive compared to CVP, right? But have you come across any data on the fact that which one is more effective? We understand we are going through CVP route, but do we have any statistical information around which one is or would be more effective?
With respect to the sample, sir?
Yes.
Sample collection?
Yes.
So there are other various studies has been done with respect to the blood serum. So which is one more, they can detect a protein biomarker as well as miRNAs. The problem with testing with respect to the blood serum is the specificity. So the biomarker can also be found in other organ cancers like breast cancer and lung cancer. Since blood is a dynamic medium, so there are chances that there will be a low
Contamination.
specificity to detect the serum. But considered with respect to the cervicovaginal fluid, it is restricted only the reproductive tract. So there will be no mixing with blood. So that is the reason that we are looking into a cervicovaginal fluid sample. But again, if it is cervicovaginal fluid sample, since it is a reproductive tract product, there is other organs like ovary and uterus cancer are also there. So that also we have verified. Is there any specificity issues with respect to the detection of cervical cancer? And so far, whatever we did, we collected the samples of other cancers and other diseases regarding the reproductive tract. And the result that we got is that in the total expression of the particular biomarker that we chose, so the total protein concentration is higher with respect to the cervical cancer than any other cancer or any other diseases with respect to the reproductive tract. However, since we have very less sample size, we have to go for more sample size. So the hospital that we previously approached has very less frequency of cervical cancer. Now we have moved to the HCG. We will be collecting more samples.
So we would be collecting more samples from them, and then again we would do the ELISA fabrication and then test it on the sensor again. The process would be the same.
Got it. Makes sense. Thank you.
Thank you, sir.
Thank you.
Thank you, team. That was a great presentation. Next, we have Team 8 Smart Shoes, Kinnari. You may begin your presentation.
Yes, yes, ma'am. Ma'am, it's showing an error. Hello. Hello.
Yes, you're audible. Please tell me.
Ma'am, it's showing an error that you have to present only PDF or photo.
Can you please send the PPT in the group so we can present it for you?
Okay, ma'am.
Yeah, thank you.
Ma'am, I have sent in that group by my technician. Can I start now?
We are sharing. Yeah, you can start now. We are sharing the screen.
Okay, so very good morning to everyone. My name is Kinti Tavde and I am here to present my project Smart Shoes for Blind Person. And my project mentor are Professor Dilipal Khambati ma'am, Professor Vishakha Khambati ma'am, and Professor Amit Mohan Raman. I am from Department of BTech Biomedical Engineering, Paro Institute of Technology, Paro University. Next slide. So begin with the introduction. So we all know that eyes are the most valuable gift that we all have. So the whole world is visible to us because we have eyesight. But there are some people who can't able to visualize all these things what happening around us. So those people face many challenges in their day-to-day life. So here I have made smart suits for them for those blind people. So in this project, ultrasonic sensor will play major role. I have I want to mention that in all blind people friendly devices, ultrasonic sensor is necessary because it is very accurate in the object detection. So this sensor is help to detect any object which is come which is come in the way of blind person. So those blind person can move easily via this smart phone. Next slide. So objective. Can you please? Okay. So this project works on the ultrasonic sensor, Arduino, battery, etc. like component. So in this project, I have used plus 9-volt battery, ultrasonic sensor, as I mentioned earlier. So this is for object detection, and battery provide the power supply. Arduino is a small breadboard in which I have attached all the components like ultrasonic sensor, buzzer, battery, and battery is used to generate power supply. And this power supply goes to the Arduino board, and after that, all components start working. And ultrasonic sensor is start the detection of any object if there is any object in the path of the blind person. So it passes signal to the buzzer, and buzzer started beeping and inform the blind person. Next slide. So this is the methodology. Next slide. So this is the detailed block diagram that I have used. So in that Arduino, buzzer, battery, ultrasonic sensor. So here I am using Arduino that 80 mega 328p module. So it is the microcontroller, completely breadboard, breadboard that is used to attach all the components, like I have mentioned that was a battery and ultrasonic sensor. So ultrasonic sensor now. So here I'm using HCSR04 module for the ultrasonic sensor. In that, 4 pins are available: VCC, trigger, echo, and ground pin. So VCC is used to power supply. Ultrasonic sensor use a 5 volt DC power supply. Trigger pin is used to used to trigger the ultrasonic wave. So it is act like a transmitter. And echo pin is used as a receiver. It is act like a receiver. So when any transmitted signal is come, so echo pin is detect that signal, and ground is for grounding the extra current. So and then battery is used to provide a power supply to the component. Now buzzer is a audio signaling device. It is also called as a beeper. And in this project, buzzer is used to inform the blind person that there is an object in the way. Next slide. This is this circuit diagram that that is how it is connected. So ultrasonic sensors and buzzer, which is connected in the Arduino board. Next slide, please. So result. So I have all components are working properly. 9 volt battery supply power completely to the all the components. So and after that, ultrasonic sensor give accurate result of the object detection, and buzzer work properly, and it produces the high pitch sound. Next slide. Now, advantages and disadvantages. So, advantages are that blind people can easily move in surrounding area while wearing these shoes. It is wearable and it is easy to use, and ultrasonic sensor give accurate result of the object detection, and person feel comfortable while wearing this smartphone. And disadvantages are that people can't cross the road, and if people is deaf, so he or she can't able to listen the sound of buzzer. Next slide. Now, application, future scope, and limitation. So application is that is used especially for the blind person, and the limitations are it is not useful in crowded area, and it is also not useful in rainy season. And future scope are that we can use advanced sensor technology to use this shoes in crowded area.
You're on mute. Can you unmute yourself?
So implement our circuit inside the shoe, so blind person can feel more comfortable. This is the picture of my project. So you can see that all the components are attached on the shoes, and ultrasonic sensor are the front of the shoe. After that, buzzer is there, Arduino Uno is there, and battery is there. Next slide. This is the conclusion. So this project is very helpful for the visually impaired person, and it is easy to use, it is wearable, and it creates comfortable environment for the blind person. This is the references. Next slide. So thank you so much.
Hi, Kinnari. Thanks for the presentation. So I'm just curious to understand, you concluded by mentioning that this is very comfortable and easy to wear. I just want to deep dive a bit on that. If I'm not wrong, the system uses 9-volt battery, right?
Yes.
That itself will add around 100 grams to the weight of the shoes, additional weight, and then the size of other components. So don't you think that would be a little uncomfortable?
Yes, that's why I'm
So
in future.
what are your reason? So I understand this is a prototype. So what is your vision to, miniaturize it, or have you thought about that aspect?
First, I want to use Arduino Nano, but there are some technical issues with that. So I have used Arduino Uno, but I am working on it to use Arduino Nano because it is smaller than Arduino Uno.
Okay. Okay. Fair enough.
Hello, Kinnari. I have one small basic question. Can you tell me the frequency of your ultrasonic sensor, what you're using?
14 megahertz.
Okay. And second thing is that you're saying that it's not advisable to wear it if you're in a crowded area. What do you mean by that crowded area? Like on road, or crowd can also be, I can understand it if
Yes.
in-house a blind person is wearing it, there'll be so many objects nearby, right? So what's your vision, how you're going to tackle that? Because there'll be a lot of objects in one household also. If the person is wearing and walking, it's not suitable for a crowded area. So how you're going to rectify this problem?
Sorry, ma'am, I didn't understand.
Okay, so my question is that you told no that these shoes are
You are
not
not
advisable.
audible.
Am I audible to everybody else?
Yes,
Ma'am, you're
Sumati.
audible to everyone else.
Okay. Now can you hear me? I guess there is some connection issue. Yeah, can you hear me?
Yes.
Okay, so my question is here that you mentioned that these shoes are not advisable to wear in the crowded area, right? So the crowd
Yes,
can
ma'am.
also be considered if a blind person is wearing it in a household scenario, right? There'll be so many things around that person. The ultrasonic sensor can detect that also. So how are you going to
Yes,
cope up
ma'am.
with that?
Yes, I have to use that another advanced technology to resolve this problem.
Okay, fair enough. Thank you.
Thank you, ma'am. Thank you, sir.
Thank you, Kinnari. That was a great presentation. Next, we have Team 16 Biomed Blasters, Shriya, Vanita Shri, Shruti Shri, Vijay Shekaran, and Vishnu.
Wait a second, ma'am. Shall I start, ma'am?
Yeah, you can start.
Hello, ma'am. Hello, ma'am.
You can start now.
Okay, ma'am. Good morning, everyone. I am Vijay Shekaran of third year Biomedical Engineering. We are the students of KPR Institute of Engineering Technology. Our team name is Biomed Blasters. I'm Vishnu K from third year of biomedical engineering.
Myself, from third year of biomedical engineering.
And I'm Shri Abhi of third year biomedical engineering. And the project guide is Dr. P. Srilata, Department of Biomedical Engineering. Just wait a second, the slide is not moving. Okay, let me go into the introduction of our project. So we know that each and every human has a different kind of approach. They have different kind of thinking. So many experiments and researchers have said that there is a relationship between these parameters like physiological signals, humans' emotional states, cognitive tasks, mental stress, and physiological load. So we propose a system which will analyze the physiological signals that has taken from an individual and which will detect the thinking state of a person and the emotional recognition more accurately and with reliability. So, as I have already said, we can calculate from the physiological signals that have been collected from the individual. The physiological signals include like electrocardiogram, electroencephalogram, electrodermal activity, temperature, respiratory activity, and arterial oxygen saturation. So for now, we are using the EEG signals to analyze the thinking state of a human. So we have collected a set of data from the DEAP data set, and we have by using those data set, we are trying to analyze the thinking state of the human, and we have done this in the MATLAB software. So the accuracy of our project is quite expected more than the already existing project that is in the market. And yeah, the following will be explained by Dimanita.
Now I'm going to explain about the methodology of this project. So there are 3 steps in this methodology. One is input, and another one is process, and next one is result analysis. In the input sections, the data are collected under some parameters like EEG, ECG, EDA, temperature, respiration, and SpO2. In this data collections, we use an EEG as an input data. So after that input, we go for an processing. So here filter processing is used. So filter is used to remove the noises and artifact in the signal. After that process, we have to analyze the results. So here we are using the 2 steps. One is signal detection algorithm, and after that analyzes the thinking state of the subject. So in this signal detection algorithm, we detect the signals by using some algorithms. After that, we have to analyze the thinking state of the patient, such as fear, emotions, etc. After that, so here is the block diagram of the entire project. So there are 4 steps. So first one is a data acquisition, is a sampling signals used to convert into a digital form. Here EEG as an input signals. After that, pre-processing method. So here we are processing some data known as pre-processing. So here we are using the 3 steps. One is filtering. It is used to remove the noises and artifacts from the signal. After that, smoothing. It is similar like the filtering process. It also removes the noises from the filter. After that, segmentations. It is the important parameters will be segmented using segmentation parameters. After that, signal processing. We have to process the signal by using either MATLAB or Python. After that, we measure the analytical state of the patient using fear, anger, or happy state. So these are the block diagrams of the entire project.
So I have already said we have collected the data from the DEAP data set. This is the data for the person who is with the sad emotion. And the next one is for the fear emotion. And next is this is for the happy emotion. And this is for the relaxed emotion. So you can see there are a parameter called a balance and arousal. So these are the 2 important parameters which by depending on these 2 parameters only we can detect whether the person is happy, sad, relaxed, angry, or anything else. Okay. So we have collected this data from data consists of 12,801 data from each patient. It consists of the emotions like sadness, fear, anger, happy, and relaxations. And from this data, we can detect the 3 types of emotion from a person like that is sadness, fear, and the happiness. So first, how we are doing is we have imported the data into the MATLAB workspace. Then by using the classification trainer learner app, which is one of the app provided by the MATLAB, we have training the models by feeding the we have collected. So the accuracies will be explained in the next slides.
So these are the models. First, we have tried co-stream model, medium KNN model, cosine KNN model, and cubic KNN model. So we have fitted our training data model that we have collected, and we have checked the accuracies of this model. So by checking the accuracies, the co-stream model has given us the best accuracy than the other models. So we have chosen this model for our project. So this is the Russell's circumflex model of emotion. So there you can see this valence and arousal as the x and y axis. So this can be easily understandable to you in the next graph. So just see this graph. As I have already said, the valence and the arousal are the very important parameters. Only by depending on it, you can detect what is the emotion of the person currently. Okay, if you have the high valence and high arousal, the person will be in the happy or the excited mood. And if you have high valence and low arousal, you will be in the relaxed mood. And if you have low valence and low arousal, you will be in the sad mood. And if you have low valence and high arousal, you will be in angry or a fear. Okay. Okay, this as I have already said, we have worked in the MATLAB. The MATLAB is a heavy software, so it took several minutes for doing our process. So within a certain given time, we can't present the total working of it. So we have recorded the video of how it is working, and we have speeded up the video for you to view it. Please.
Now I want to explain how we did the project using MATLAB in this short video. First of all, we train the data set. Data set is nothing but it consists of data. Totally, it has around 15,000 data. We separated it 12,000 for training and 3,000 for testing purpose. After training the data set, we can see the accuracy level in this year. Next, we exported the model. Then the function will be generated in display. Next, we imported some data, training data. And we saved the data of the training data. Like training data, we exported and imported data of the training data. Next, we evaluate the function like step by step. At last, we can see the accuracy level here. Here you can see the accuracy level of the data. Next, I go to Sri.
Okay, what we have said is first, we have trained the model. Actually, we have collected around 15,000 data. So in that, the 12,000 data has been separated and we have used it for training the model. So in the training model, we have this arousal and the valence values as default. And by training the model, the model will be trained according to the data that we have given in the training model. And then after training that model, we are importing the testing data, which doesn't contain this valence and the arousal values. So only by using those energy, entropy, and SD values, the valence and the arousal values will be calculated by the model itself, because it is already trained by using the training data.
Applications. The emotion detection, we can apply this in a safe driving or healthcare. In healthcare, we can apply this for a coma patient. We didn't know how they will respond. They cannot talk to them. They cannot do any actions, right? We have to know their emotions. Means we can fix this EEG in the EEG to the coma patients, and we can detect their emotions. And most of the peoples now are stressed.
sad. For most of the IT companies, they have a low workload, right? We cannot say by their facial expression they are depressed, stressed. If we made this product into a cap, it will be user friendly. So it will be useful for the workers and the people who always stressed. So by this we can detect they are stressed and we can improve our health. And this will be useful for the psychologist. And the limitations for now, we can use this only in a labs. It is not that much portable. If you made it in the cap, it will be portable, user friendly. Over to.
In a MATLAB software, we are using in MATLAB software we are a number of apps. In apps, we are using a classification learner. In classification learners, we are a number of models, but we are trained just only 4 models, like quasi KNN, cubic KNN, medium KNN, quasi models. But we are getting an accuracy rate, more accuracy rate only in quasi model. We got less number of accuracy in other models. The accuracy rate of quasi is 99.68%. But next, the conclusion is in a MATLAB software we are importing some data. In data are only in data set there are parameters like sad emotions, fear emotion, happy emotion, and anger emotions only. We can predict our sad, fear, anger, happy through valence, arousal, and electrons parameters which are required by our code, and we can improve our product by using the software as a cap or as a product, and now we can only use in the laboratory purpose because it is not a portable one. So this is the main drawback for us. In future, we can make this as a portable one, and we can include a more this we can display in the screen by saying you are in a sad, you are anger, and we can intimate that you are in by displaying or glowing some bulbs, some alert messages like. And these are the reference idea we got these are the reference we got an idea for this project. Thank you.
Sir, ma'am.
Shubham, you are going to ask?
Yeah, please go ahead.
Okay, thank you. So the first question, guys, that you told that you are you have taken 50,000 data set.
15,000, ma'am. 15,000.
Let's say 15,000. It's a real-time patient data, or you have taken some library?
We have taken from DEAP data set, ma'am.
It's a library, online library you have taken. Okay. Okay. And second question is that
created from the patients, ma'am. That has been taken from the real patients.
But it's an online database.
Yes,
right?
ma'am. Exactly.
Okay. Can you tell me how much time it took you to run all 15,000 data set?
It's not quite tough, ma'am, because we are not running the whole data. We are just importing those into ma'am. So it takes almost around 1 day to run the whole data, ma'am.
Okay. Tell me what will be the possibility of error, because you're telling that you're taking the psychological data sets, right? Your ECG, EEG, SpO2 signals basically.
We are only taking the EEG signals now, ma'am.
Yep.
Okay, let's say you're taking EEG signals for now. Then in that case, EEG signal can also be, there'll be a variation, no, in case of any underlying medical problem also. That can also be a cause of signal variation. How will you manage that it is not because of any medical condition and it is because of the, like, maybe a person is showing variation in peak. As per your algorithm, it will tell that they are happy, but
how much it will be sensitive, that maybe they have some underlying medical condition also.
Ma'am, it is only for the normal people who are working, who will be checking for, ma'am.
Yeah, but you're saying that this future aspect of the product is to use it in coma.
Yes, ma'am. Because there will be no much variations in this EEG, ma'am. Just by taking the EEG of this coma patients, we can get this data from their waveform and we can easily do it, ma'am.
You're
There
saying
will be
that
no much the accuracy will be very good, I mean.
No, but it can be due to some underlying medical conditions also, right? So how sensitive your algorithm will be? What will be the sensitivity that if the person, let's say, actually happy and showing happy? What will be the sensitivity? Have you got any error data till now?
No, ma'am, we have not got any error right, ma'am, but we have got some errors, but we have corrected it a lot, ma'am, in coding, in the coding of the MATLAB, ma'am. We have got first less accuracy when we are coding, ma'am. So we started running the program little bit. It started to give the good accuracies, ma'am.
Okay, as per your data set which you have presented just sometime before, as per that you're mentioning that using the cross tree you have received 100% accuracy. It's making
98.85 percentage.
Okay, so what do you mean by that accuracy?
That accuracy is when the valence and the arousal values of the trained data, we have already fed into it. So when we give the testing data, that is only by giving this energy, entropy, and standard deviation, it itself gives the balance and the arousal as like it is in the trained data, ma'am. So to be frank, we have not made to give an output like whether they are happy or sad, but now we have made to give the values, valence and the arousal values like how it could be, how it could be for these energy, entropy, and SD levels. So based on this arousal and balance values, we can detect whether this person is happy or sad or fear or not.
Is there any chance of error that out of those 15,000 data, let's say, you have just calculated your balance and arousal, right?
Yes, ma'am.
So how you're gonna, is there any chance that these values can be intermediate values also? It can give you error in terms of...
we are not taking only set of values, ma'am. When we are taking an EEG signal, we take we will take lots and lots of sets of same parameters for a specific interval of time, so that we can take a set of values and we can approximately give the, but by taking the approximity of these values, we can give the whether the person is happy, sad, or
Okay. So what's your future vision? Is it going to be a real-time detection that you will connect it directly? There'll be some app or something?
on it, ma'am. Like we can place these electrodes in a cap, or there are lots and lots of ways to detect the mood of the person. Like we can made a sensor like to check the skin resistance of a person. By using that, we can make a watch just similar to this project, and we can check whether the person is in happy mood, sad mood, or something else.
That's fine. That's fine. I am asking you. You are using EEG. What will be the vision for your device? EEG cannot be done in the form of watch, right?
Yeah, ma'am. We can make into a cap, ma'am. Mainly, we are thinking to by placing the sensors there, we can make a by miniaturing the size of the particles which are using, and we are making a easy and comfortable cap so that the persons can use it easily, ma'am.
Okay. Thank you. That's all.
Hi. So as accuracy is already as high as more than 95%, why do we need any other vitals? What's the need of that? I understand we worked on EEG to get to this result, but why do we need any other parameters then? So ECG, EDA, temperature, you mentioned about SpO2 as well.
So we testing only ideal conditions.
Okay. So is there any plan, and I guess that would have answered one of the queries of Sumati. If there are any other complications, how do you narrow it down? How do you not know that it's a false positive? So probably getting more data points can be a better way to, narrow down and be more specific. As simple as if someone has high temperature, that can mean something. And then you can combine it to get a better signal. Okay, well, that is all from my end as well. Thank you.
Thank you, Biomed Blasters. That was a great presentation. Next, we have Team 17 Stallions, Fatima Hakim and Rosemary Tommy.
Yeah, good afternoon, everyone. We are here. Shall we present?
Yeah, you can present now.
Hope the PPT is visible, right?
Yeah, it's visible.
Yeah, so our project name is Intravenous Infusion Monitoring System. And myself, I'm Fatima Hakim, my companion, Rosemary Tommy. We did this project under the guidance of our professor, Sajida S. And we are from Government Model Engineering College, Trikakara, which is in the state of Kerala, under the Department Electronics and Biomedical Engineering. And we are from third year. Over to Rosemary.
Next slide. Good afternoon, everybody. I'm Rosemary Tommy. So the project that we are currently working on is called the Intravenous Infusion Monitoring System. So fluid administration as part of treatment is a crucial component of the hospital, which needs continuous monitoring. So it is quite challenging for a nurse to keep an eye on each and every individual. So in order to ensure patient safety and for them to reduce the workload, we developed an alert system that gets activated when the fluid level goes below a certain level, becomes obstructed, or if any motion is detected. Next slide. Next slide. The objectives of the project are, first, if the fluid level falls below a certain threshold that is set by the user, the alert system gets activated. Second, it identifies any obstruction in the fluid flow. And third, it senses the hand's movement and any accompanying blood recirculation. Next slide. So the above diagram, so the picture that you can see on the screen is a diagrammatic representation of our project. The blue color cylinder represents the drip bottle. So above the drip bottle, we have placed an ultrasonic sensor which continuously measures the fluid level. So if the fluid level falls below a set threshold, the alert system gets activated. And if the level of the fluid remains constant for more than 3 minutes, even that gives you an indication. A PIR motion sensor is placed very close to the cannula to detect the motion of the hand. So if the person moves his hand very vigorously, it can cause the needle to get displaced. So if the needle is displaced, it can cause the backflow of blood into the tube. So in order to check for the presence of blood in the tubes, we have an IR module across the tubes. So the information from all the sensors are given to the Arduino, that is our control unit, where the data is carefully analyzed and the results are then given to the nursing station in the form of visual indication and audio indication. Next slide. So the components we have used is Arduino Uno R3. So Arduino integrates the information from all the sensors and carefully analyzes the data and gives you the corresponding output. So in our case, the output is given in the form of LED. That is, LED gives you the visual indication and the buzzers will give you the audio indication. That is, this alert can inform the nurse on whether any faulty situation is occurred. And HCSR ultrasonic sensor measures the distance, and the distance measured using ultrasound waves. So the distance in our case, it measures the level of the fluid, And if the fluid level falls below a set threshold, it means that the bottle should be replaced immediately. And the third one is the PIR motion sensor. So this sensor detects the motion of the hands or the organ of the body in which the cannula is being placed. So this is done with the help of the IR rays that is emitted by the object. The IR module includes the IR LED and the photodiode, which checks for the presence of blood in case any backflow of blood has occurred. Next slide. So Fatima will talk about how we implemented our project into a working model.
Yeah, so first we did with the software analysis. For that, we used the Tinkercad. By using Tinkercad, we developed the circuit for ultrasound sensor and PIR motion sensor. And in the software version, it worked very successfully. And for IR module, this was the circuit diagram. We used an IR LED and photodiode. IR LED emitted the IR waves, which was detected by the photodiode. And we have a short working video over here. So shall I play the video?
Yes.
Now we have uploaded the code for PIR motion sensor. This is the motion sensor. If we move it, LED blinks. Otherwise, the LED will be off. Now this is the circuit using IR module for detecting the blood backflow. Assume this to be the blood. When it shows, the LED blinks. And when it is taken away, the LED becomes off. And now this is the circuit for detecting the fluid level in the bottle or drip bottle by using an ultrasound sensor. The sensor has been placed at the top of the bottle, and since now this bottle is filled with the fluid, the alarm is off and the LED is also off. When the fluid level drops between these two red lines, the LED blinks and the alarm turns on. So now let's see that. You can see that the water level or the fluid level has dropped between these two red lines, and now we can place this ultrasound sensor. So now our aim is to detect is there any blockage in the drip level. So it is checked by the following method. If this level remains constant for more than 1 minute, the alarm and the LED blinks. So let's see.
Yeah, so this is a working model of our project, and this level detection, the constant we now we set for the 1 minute. It is for the easiness of recording the video. In the program, we can make it to 3 minutes. So if the fluid level remains constant for more than 3 minutes, it indicates that there is an air blockage or air has trapped and the fluid is not moving. And so coming to the results part, we were able to achieve all our objective. That is, when the fluid level fell below the threshold, the LED blinked and the buzzer sounded. And when the fluid level remained constant for 1 minute, it can also be set as 3, 4, or 5 minutes, depending upon the easiness of our. The alert was issued. And when the hands or body part, that is to the body part to which the motion sensor is attached, when the part moves, the LED blinks. Whenever there is a backflow of blood is detected, the LED will be blinking. Application, as we said in the problem statement, every hospital needs a fluid management system, because whenever we go for a disease, let it be from small to big, the primary treatment is giving a glucose treatments or some invasive injections into the body. So it is very difficult for the nurses to randomly run to all the beds of the patients and monitor whether the level is about to over or not. So this brings a cost-effective and efficient method in the hospital to decrease the workload of the nurses and ensures the patient safety. So that itself is the advantages. It ensures the patient safety makes it more easy for the nurses, and carelessness due to nurses can be avoided because in every hospital, right medications in right dose and all is very important. The one disadvantage we found here is in case of IR module, it is very ambient to the, it is very sensitive to the ambient red light too. That is the only disadvantage we found. And future scope is that currently, this is the prototype we have presented here. In future, we can integrate an LCD display so that we can correctly indicate the fluid level, also displaying the fluid level. And another one is we can set an automatic flow rate setting also. These two can be integrated into our project. This is our future scope. These are some pictures of project. These are initial level when we introduced module wise testing, and this is how now our project looks like. It have PIR sensor, ultrasound sensor, and IR module too. So coming to the conclusion, the project assisted in the monitoring of the intravenous fluids. We were able to achieve all the objectives that we mentioned. Alarm was given, and we are very happy that our product or our prototype can assist in the hospitals, ensuring patient safety as well as can bring smiles on the faces of the nurses and patients. So these are the references that we used. And that's all about project. Thank you.
Hi, Fatima. Hi, Rosemary. Thanks for the presentation.
Yeah.
Thank you, sir.
So I was briefly disconnected in between. So I just heard the initial part of it. I just wanted to understand why we are only sticking to IR sensors. And I think in one of the initial presentations, Sumati did talk about the limitations and disadvantages of IR sensors. So how are we so sure that this IR sensor will be capable of taking care of all scenarios?
Yeah, we mentioned that in our disadvantage also. IR sensor may be sensitive to ambient light also. We actually looked for a color sensor that is TCS 3200. But when we were prototyping it, it was not available. So that for the time being, we just used the IR module. We have another option also, RGB color sensor that is TCS 3200.
Okay. Okay. So wouldn't system be slightly different if we are changing it with those sensors?
The question is not clear. Could you repeat it?
Yeah. So if we use those color sensors instead of IR sensor, wouldn't the system drastically change? Or do you think the similar parameters would be relevant?
Yeah, there will not be any change because the TCS is also basically a color sensor. So just need to code it. It will be detecting the red color.
Okay, got it. Fair enough. Thank you.
Thank you, sir.
Hello, guys. I have just one basic question. So what will be the difference from the manual infusion pumps which are already available in market and the one you are developing?
Yeah, that's what we say. When in the manual infusion pumps, the nurses have to keep on checking whether this is about to over, and she have to go to the nursing station, come to the patient, go to nursing station, come to the patient. I have to sit throughout with the patient. But in case of this, we are sending alerts, so she can sit in the nursing station, or even though if she is any other ward or in nearer in other beds, whenever she gets an alert, she can come soon to this patient and can remove this cannula can be disconnected.
Okay. And second question is, what are the options you're looking for in terms of sensor? I guess Shubham has mentioned this thing. So what are the other options you're looking for, if not IR?
That is the same thing. That is TCS 3200. It is a color sensor, RGB sensor.
Yes. Okay, that's all from my side. Thank you. Thank you, guys.
Thank you, ma'am.
Thank you.
Thank you, Stallions. That was an excellent presentation. Now we'll be taking a break and we'll be resuming at 2:15. Thank you all, and please do join back at 2:15.
Class line meeting.
Okay. We'll be waiting for another 5 minutes as there are only 17 participants. Tarniraj, you're ready, right?
Yes, ma'am.
Okay, okay. We'll wait for another 5 minutes and then you can start.
Okay, ma'am. Okay, thank you.
Thank you.
We'll be starting shortly. May I know whether the judges are ready?
Yeah, we are ready.
Yes, fine.
Yeah, okay. Tarani, you can start your presentation.
Yes, ma'am. Thank you, ma'am. Yeah, very good afternoon, everyone. This is Dharani. My project is My Name Sarah, means St. Philomena's Employee Robo Attendant. Team name is Maya Mantra, name of the engineer Tarani, project guide Tarani, Department Biomedical Engineering Department, name of the hospital St. Philomena's Hospital, Bangalore. Introduction. The peak of the pandemic and the highly infectious disease will pose serious threats to both medical workers and other hospital patients. To counter this, while protecting our healthcare workers and other hospitalized patients from unnecessary exposure, by allowing medical workers to remotely help the patients using Sera remote control robo with self-drive. Sera remote patient monitoring significantly reduces the frequency of contact with the suspected and confirmed COVID-19 patients, thereby protecting the staffs and technician and reducing the patient requirement for PPE. Objective. Sera can kill the virus at empty room by switching the UV rays. Sera can spray the hydrogen peroxide at empty room by switching the sprayer. See, method is Sera is the semi-autonomous robot designed to support COVID-19 patients and frontline workers. Sera robot built and designed to engage in hospitality management and workplace productivity, reusing the condemned wheelchair and making it working condition, and that wheelchair converting to the electric wheelchair with the joystick optional, developing electric wheelchair to remote control access. So wheelchair wheels run on 24 voltage permanent magnet DC GAD motor and aluminum alloy frame with powder coated finish, reclining back to maximum comfort, and safety belts for added protected, and car seat designed for maximum protection, and anti-toppling wheels to avoid accidents. Detachable elevating footrests equipped with a touchscreen with joystick and radio frequency remote control. Once charged, could last up to 8 to 10 hours. Sera robot can be used all COVID-19 wards and ICU at St. Philomena's Hospital. So main project block diagram is AC input to the inverter, and it will go to the charging station. Battery will get charged, and same we will get the AC output for the utility. Example like camera, UV chamber, siren, sprayer, and from inverter it will come to the driver board. As transmitter what command is giving to the RF receiver, it will go to the driver board, and that control it will go to the servo motor relay board, and it will activate the right left motors. So joystick is optional here. So during implementation, receiver servo motor converting to relay switching was little difficult, and we have done it. During the project, so previous slide, please, sir. Yes. So during this project, permanent magnet DC motor placement and matching required wheel speed with soft RPM with high torque was the difficult. Matching right left facing the motor direction and matching the RPM was the difficult. So what we got result from this project, doctors can talk back to concerned nurse or patient directly with using two-way talk-back communication system. Doctors can access the particular patient data remotely in real time, and staff can deliver the medicine using Sera. Staff can deliver the meals using Sera. Sera can transport equipment such as pulse oximeter or infusion pumps, syringe pumps from A ward to B ward. So apply a Sera remote control a robo with self-drive. That's a major application for us. So we fulfilled maximum advantage with this project. We sprayed visual effect against coronavirus silver nanocluster silica composites butter coating for the Sera robot. So we called as like zero virus. Okay, it will validity will be 1 week, and we used MI 360 camera Wi-Fi. And we used drone technology 2.4 gigahertz 6-channel transmitter. And in this project, the disadvantage is this not waterproof, and less our battery back, like only 8 hours we can get it. And one more disadvantage is knowing charging status is difficult, because remotely we are accessing, we cannot able to see. So camera angle only 21 degrees angle. So in that circuit we cannot able to see. So that was the disadvantage in this project. So application we can customize. This is we can dismantle and we can reassemble. We can change the model, design, everything, and we can upgrade to the management mobile surveillance in future, and the campus daily rounds and observation and measuring the environment, air purity and temperature. So firefighter with cylinder extinguisher, it's possible. And limitation autonomous wheels to check safety points status, daily checking like GPS status and power status, gas pressure level, and line checking, and face recognition system like a stop or any other ID can be modified, and real-time digital data storage can be done using this project. That's a possible. So this is our project picture, and right side is the specification. Okay, and above top is the MI 360 degree Wi-Fi camera, and down siren is there. So anyone's coming near to project and touching or disturbing, so we can activate the siren or any emergency. And wheelchair seat is like multiple purpose, multitask. We can use it. Even sick patient can drive with the joystick, and we can monitor, and the GPS is there, and the left side hydrogen peroxide sprayer is there, so we can spray, we can sterile the room where we want to sterile the room, so we can do it. So we are using that. So this conclusion from our management side, like administrator, medical director, finance manager, and nursing superintendent, they said that we conclude that on the basis of our hospital market and survey, we made a best design of robo. We automate means remote controlling, forward, backward, right, left motion. The benefits of the project Sera is to maintain and customized usability, easy access with remote, durable and chargeable, high strength and elegant design, and cost benefit. Sera make it possible for patient to perform tasks without help from other people like technician, therapist, nursing staff, and a good communication carrying real-time Sera solution can ensure better patient safety while safeguarding staff and doctors against exposure. So clear clinical accuracy, affordability, and above all the ease with which we can go live and get the right timely patient to save lives. So YouTube video is there, so that link. Thank you very much for everyone and team and participant.
Hi, Tharni.
Yes,
Thanks for
sir.
the presentation. So when you mention semi-autonomous robot, so what does
Okay.
that mean? Will there be a pilot required all the time to navigate it?
True, sir. True. Exactly. Someone has to give command using that RF remote control sitting in any other place. Okay, they can command by seeing the video directly. So how they like it's a drone on wheelchairs. That's the concept actually. So how we are using the drone instead of drone, we are using the wheelchair here.
Okay. So why
Yes,
don't we
sir.
use drone? I'm just exploring that option as well, as you already mentioned it. Why do we
Yeah.
want it to be grounded?
Yes. So if drone means there is some we can't carry any one pulse oximeter, so that balancing and gravity and so a lot of things is there. For our focus on helping the COVID-19 patient. Okay, they comfortably they can talk to their relation. Okay, so a lot of multitask method. So that's what's in drone. It's not possible actually. Drone
Right.
only just we can go and we can see. It will not helpful, but just we can see that. That's it. But
Fair enough.
yeah.
But you do realize the COVID scenario was one of sort of an exception than a norm. So if we have to make a point that this is still useful post-COVID era, right?
Okay.
So you did mention a couple of applications. What do you think would be the best use case of such a system?
Okay, this management mobile surveillance we can do it, sir. Like every day, like we can this now semi-autonomous we can convert to autonomous, and we can with using GPS and Google system we can automate. Okay, no need to control any pilot. Automatic daily checkup done with using the safety measures. So it will not at any walk-ins or any other people. So like that we can program in future. So what are all safety measures like humidity, temperature variation, or dog center, or rat find, or a different? We can program it, sir. Like some emergency also, it's automatic fire system. We can configure with this. Any alarm, any fire alarm, it will reach that particular place and it will observe any thing is there. Then fire is there. It will blast the fire extinguisher and it will use. So like then we can program.
Okay.
Future.
So it looks like you want to make it a Swiss knife. I'm quite intrigued, but at the same time, what is the maximum payload capacity that it can handle?
Okay. In this, sir, for we want to make it user friendly for the patient. So that's COVID-19. It was very difficult. So we want to help frontline workers and doctors. They were struggling for the collecting the data for each and everything. They want to enter into COVID-19 ward and they scared anxiety, fear. They were doing work. We understood everything and keeping in mind. So we have made this project, sir.
Yeah, that's true. What I'm trying to understand is what's the maximum payload or the weight it can take and then carry around.
Okay, maximum 80 kg, sir. 80 kg.
80 kg. And then with that 80 kg, it is able to operate for 8 to 10 hours continuously.
Yes, of course. Okay. Yeah, correct. Yeah.
That's interesting. Got it. Thanks.
Okay. And one more important thing I want to mention in this. Okay, we were using sterilization for room. We were using the fumigator and other things. So in this, we changed that system is like hydrogen peroxide during COVID-19. So still now also we are using. If no need to get any fear, just we can send and 360-degree we can able to see that how that spraying or what. We can switch on and we can see. No need to enter anyone. So one more future advantage is like by holding that remote control, we can operate the radiology department. Like we can expose the X-ray machine also, stand by, and we can shoot that also. It's possible, but we are not done. So future we will do it, sir.
Okay.
Yes. Thank you, sir. Thank you, everyone. Yeah.
I have a question that you mentioned that you're using UVC for sanitization purpose, I guess.
Yeah. that ultraviolet rays for shifting the patient. Patient is recovered and we want to sterile that room. After shifting the patient, we will use the ultraviolet rays for
So,
killing
but
the virus and bacteria.
yeah, but what's the range then you're using the UVC? Because if the patient is already present over there, the UVC should not be harmful for the patient.
True, ma'am. 100% true. After evacuation of the patient, then we will send this robo inside and we will see each and every corner. It will rotate, ma'am, actually. If all is big also, no need to worry. We will send to corner and we will activate the UV rays. Then that bacteria, any virus, it will kill.
Okay, what's the range of UVC you're using?
What UV rays, that's small size only, 4x4 range, ma'am. 4x4.
No, this is the size of UV module. What's the range of wavelength it is? Like for
Wavelength.
sanitization, let's say we use 275 nanometer, let's say. In case you're sanitizing some equipment. So what's the range over here?
Yes, ma'am. In this 150, I guess, ma'am. I'm not sure 150. Minimum range only we were used actually. Minimum range.
Okay. Okay. And second question is that you mentioned that there is a motor present which is triggering the movement of the chair, the robo, right?
Yes, ma'am. Yes. Yes, ma'am.
So which type of motor you are using?
Permanent magnet DC motor, 24 voltage we were used. And in this same motor, that time market, it was not available. Or we purchased the same configuration, two motor. So we have to place it in wheelchair backside, right, left. If you are placing the motor same direction, same facing, then RPM will differ, ma'am. For matching that, again we have to do some modification. So we have to rotate, we have to measure that RPM. So one RPM changing also, it will not go straight. So that's what we observed during the project. So it was difficult for us, and we modified some technical things, and we have done it now.
Okay, and the third question, Thani, is that you're saying that it can be operated through remote.
Yes.
What are the functions that will be available which are available on the remote?
What we are used for this project is Fly Sky 6 channel 2.4 gigahertz. So that means the 3-kilometer radius we can able to control anything, ma'am. It's a possible. Like drone you are controlling 3-kilometer distance. Why not we can't control inside the hospital campus? Okay, main entrance of the hospital with sitting my office I can control. I can open and I can close. I can control in 3-kilometer radius, and I can activate the X-ray machine using that servo control motor. I can control, ma'am. It's possible. We have done it.
Okay, okay. That's all, Thani. Thank you.
Okay. Thank you, ma'am. Thank you very much.
Thank you, Tarini, for that wonderful presentation. We next have Team 12, Innovative Engine, Yuvraj, Priyadarshan, Shvetashri, and Soumya. Is Team Innovative Engine ready?
Yes, ma'am. Yes. Soumya, can you share the screen?
Yeah. Okay.
Okay. Good afternoon to one and all present here. Myself, Priyadarshan, and my other teammates are Soumya, Shvetashri, and Yuvraj. Yuvraj could not attend this meeting because he's busy with the internship courses. And we are studying in Agni College of Technology, which is affiliated to Anna University. We all belong to the Department of Biomedical Engineering. Our project title is Cost-Efficient Scalp Cooling System for Chemotherapy-Induced Alopecia Patients. Our main aim of the project is to prevent or reduce the hair loss for cancer patients during chemotherapy treatment. Our objective of the project is applying cold temperature to scalp during chemotherapy treatment for cancer and to provide cold cap at affordable price. Presently, if you see the cold cap price is about $1,000 to $3,000. We are affording it at just $180. We are affording it. and to reduce the amount of chemo treatment that reaches your head, protecting your hair follicles from chemo drugs. By this process, we will not allow the chemo drugs to reach our hair follicles, which prevents the hair loss for the cancer patients. Over to Soumya.
Methodology. In this process, we are reducing the temperature of the scalp. It reduces up to 40% of the medicine that reaches into the scalp. We use technology to reduce this temperature of the scalp. For this, we use Peltier effect. Here's the diagram of Peltier effect. It gives a cold side, it observes cold in one side and rejects the heat at other side. You can see the diagram. And the implementation. This is a block diagram of our model. In this first, the switch mode power supply converts 230 to 240 volt AC power supply into 12 V DC power supply. The tech cooling system starts working, then the thermostat controller starts sensing the temperature of the aluminum block, which is connected to the Peltier, and the buzzering is started to buzzer when the acute temperature attains the level. Now the water pump naturally pumps the water into the container to the aluminum block, which is connected to the Peltier effect. Now the cooled water reaches the scalp, cooling temperature of the scalp, reduces the scalp temperature and reaches back to the water and cold water container. This cycle continues till the end of the chemotherapy process. Over to Shweta Sri. Hello.
Hello.
Over to Shweta Sri.
Hello.
Yeah, please
Hello.
continue. You're audible.
Yes, you're audible. You can continue.
Okay, from this results, we can see that hypothermic caps can lower the bladness of the hair loss, and it can treat the alopecia patients while during the chemotherapy treatment. Next, here is a video about our project, about a working of a project. Soumya. Next, we can see the advantages of this equipment. Cool. Advantages is this equipment is lighter than the existing model, and this is a cost efficient model. It is a eco-friendly model. This does not release chlorofluorocarbon, since because the existing machine will release the harmful gases like CFC gases. This size is very compact in size. It's very easy to use. Nowadays, the patients having the queries that they are not comfortable in wearing this cool cap system. So we have made this model in a very efficient and eco-friendly method. Next is the disadvantage of this model project. Our project is this won't suit for the people who are suffering from sinus. This leads to the cancer patients who are having a sinus problem, will cause other health issues like forming cold, sinus, and other. Next slide. Future scope of project is we can set a digital timer which can automatically switch on and off of our equipment. We can we can use it can be developed into two. We can set two caps in the we can set two caps in the one system, because it can treat two patients at a single time. This only save time but also saves money to the patients, because the existing machine is about $8,500 to $3,000. So our machine is just $150. Next, this is the complete picture of a project. Finally, we conclude that the scalp cooling system reduces the hair fall to the chemotherapy patients, chemotherapy induced patient, alopecia patients. It also reduces the temperature, resulting in the blood flow to the scalp here, so it lets the chemotherapy drugs such hair cells. Next, these are the papers we have taken for reference for this project. Next, Soumya will share the promo video of our complete project.
Yeah, this video is not.
Try downloading it and then play it in the browser. That might work out. Download the file itself. Anyways, meanwhile, I have a couple of questions that I want to go through. So going through that presentation, one thing that I was able to decipher that cap or that entire system is acting like a heat sink. It will take heat from one end and then move it to the other end. Have we tested it even on a normal person? Forget about a chemo patient or during the procedure. Have you tested it? Have you even measured the temperature difference that is being produced?
Yes, sir. But we have not tested this project on a subject. But we have gone through this, and we can control the temperature through the.
Sweta, you are on mute.
No, sir. Till now, we didn't choose any subject and we didn't test, sir.
Okay. But how are we so sure that it's going to work out then? Do we have any evidence?
It's already in market, sir. They are using the 20 to 15 degrees Celsius of cooling, sir, in the subject.
Okay.
So we are used to reduce the cost, sir. In this, we are providing our mission in affordable price and time saving. It is a continuous mode, sir. In previous mission, there is a If we are using a cold cap at 40 minutes, then they wait for another 1 hour to cool the cap, sir. But in our model, it is a continuous model, so it cool the system continuously till the patient gets the treatment.
So what's the temperature difference we are able to get from the
15
up to so it drops till the 15 degrees Celsius.
15 to up to 20.
Okay. Got it.
Hello guys. So I have the same question in continuation. What's your input temperature and what's your output temperature?
No, ma'am, there is no input and output temperature. If we pour the water into the water container, ma'am, the peltier is started to cool the system, so it provides 15 degree to the patient. It reaches the scalp and it enters into the water container again, and it again cools the water and it again reaches the scalp, ma'am. It is continuously moving the water in cooling.
Okay.
Process
Now
takes place continuously, ma'am.
Okay. Now the second question arrives here that for cooling you're using some metal.
Yeah, aluminum block, ma'am. We are using aluminum block that is placed in the peltier. So the peltier cools and the...
So the question here is that how you're controlling the earthing? There can be a
thermostat controller, ma'am.
Temperature using thermostat controller.
Okay. So using the thermostat, you're controlling the temperature.
Yes, ma'am.
Yes, ma'am.
Ma'am, we are controlling the speed of the water is through a potentiometer, ma'am.
That's fine. My question here is that to cool the water, which is a conductor, a metal you're using, which is a conductor, right? And there is some power you are giving it to that conductor to cool it basically. Right?
Sorry, ma'am, can't understand the question.
How your metal is getting converting your temperature? Everything is connected to electronics,
Converting
right?
through Peltier model, ma'am.
That's what my question. That your full circuit, have you
Then
there is
yes, ma'am.
at any point you have thought about earthing? Because you are directly going to use it on a patient, right? And it is connected directly to an electronic circuit.
No, ma'am, we are giving 12-volt power supply to the peltier, ma'am.
That's fine, but a person can get a static shock also, no? Static current also.
No, ma'am, the patient won't affect through this, ma'am. Electrical, it won't cause electric shock or somewhat.
Okay, let's say if it does any problem or it shorts to your actual temperature. That's why I asked my first question. What's the control temperature you are going to take?
It's 14 degrees Celsius. Ma'am.
It is 14 degree. Then how much lower it is going?
No, ma'am, the last is 14, ma'am. It's going higher to 20, ma'am. 20 to 14.
Dear, I'm asking that let's say you're pouring some liquid, let's say water. It will have some temperature, right? So that means if you're using it on the room temperature, it is 37 degrees. From 37, is it going to 14?
No, ma'am, 37 to 14, ma'am.
That means from room temperature, it is cooling down to 14.
Yes, ma'am. From room temperature, it is cooling, ma'am.
Okay. Thank you.
Thank you, ma'am.
Thank you.
Thank you, Team Innovative Engine. Next, we have Team 19.
Ma'am, one second. One second, ma'am. Here's a complete video. Yeah, sorry, ma'am. You can continue.
Team Innovative Engine. Okay, fine. Thank you, Team Innovative Engine. Next, we have Team 19, Health in News, Meenakshi. Is Meenakshi here?
Yes, I'm here. I'm sharing my screen. Just a minute.
Okay.
And the spinal cord injury paralysis is most often caused by especially in the spinal cord. This means that the nerve signal sent to the muscles is being interrupted. Paralysis can be broken down into two types based on the site of injury in the nervous system, which is flaccid when the muscles get flappy and shrink, or spastic where the muscles localized affect the small sections of the body, mostly the brain and feet of the heart. Generalized paralysis affects a larger area. This monitoring system helps patients to convey their messages to doctors, nurses, and their loved ones easily. This monitoring device helps patients with only partial paralysis to convey their messages in emergency. Next.
Good afternoon, everybody. I am going to be speaking about the objectives. In this project, we have tried to cover the most essential gestures needed for an easy communication. For example, when a patient moves his palm towards the right side, the corresponding messages gets displayed on the LCD with the buzzer beep. Similarly, this happens for a few more hand gestures. By developing this project, the health officers can assist paralyzed patients when they are alerted by messages from the patient. This project is designed with several instructions or gestures, which helps them to manage their daily needs, such as drinking water, eating, etc., which alerts the health officers to assist the patients to complete their needs. Whenever a patient gives a simple hand gesture or instruction, the corresponding message from the transmitter will be delivered to the receiver, and the message is displayed on the LCD receiver. There is also a buzzer that provides an alert to the health officer simultaneously with the display message when the officer is not in the closest vicinity of the receiver. The components consist of a transmitter and a receiver. The transmitter consists of an accelerometer of GY61, an Arduino Nano voltage regulator 7805 voltage regulator is being used to convert 9 volts to 5 volts. An encoder HT12E is the encoder. We use this to encode and transmit the signals. The components in the receiver is a buzzer, LCD display, Arduino Uno, and a decoder HT12D is used to decode the signals received. The next will be taken by Meena.
Okay, good afternoon, everyone. Starting with the implementation, basically we have the transmitter and the receiver sides in our project. So on the transmitter side, whenever a patient needs anything like food or water, etc., they can move their hand, and this movement is sensed by the accelerometer, which is in the transmitter that is mounted on the glove of the patient, which will be worn by the patient and sends the signal to the Arduino Nano. So, and the power supply given to the transmitter is 9-volt battery, which is converted to 5-volt supply by the voltage regulator 7805. The signal after being reduced in the Arduino Nano gets sent to the encoder to encode the signal and transmit it to the receiver. On the receiver side, after the signal is received by the receiver, the HT12D decoder decodes the signal and sends it to the Arduino Uno. A USB is connected to the Arduino. There are a certain set of messages which will be displayed accordingly to how the patient moves his or her hand. After receiving the signal, the buzzer makes a sound indicating the patient's needs. or if there is an emergency, and this will be displayed on the LCD display screen so that the doctor, nurse, or loved ones can provide help to the patient. Coming to the flow chart, first the hand gesture is recognized, which in the transmitter, and then it is sent to the receiver. In the receiver, it gets decoded and sees if the hand is turned to the right. If yes, it displays a message on the LCD that I need juice. If no, it goes to the next step, that is the hand raised up? If yes, it displays that I need water. If no, it goes to the next step, that is the hand facing down? If yes, it displays I need food. If no, lastly, it checks is the transmitter facing the ground. If yes, it displays take me to the restroom. When all these are getting displayed simultaneously, the buzzer is on. The message is displayed on the LCD and the buzzer gets off and the process stops. Next will be continued by Mansi.
As you can see in the picture, there is a transmitter and a receiver. The transmitter is situated on the glove, and the receiver is on the doctor's side. When the hand is turned to the right side, the message that is received by the doctor is I need juice. And when the hand is facing upwards, the message is I need water. And when the hand is facing downwards, the message is take me to the restroom.
Advantages. If any health treatment, the device is the disadvantages are due to handling in real time, and there could be risk of or even power failure. Healthcare operations. Only fully paralyzed people cannot use the device to produce any gesture. Applications are this device can be used to comfort take doctors hospitals or communicate with doctors. So asking others taking care of patients without the loss. Conclusion is the device was made giving back or communicate. It is low cost and reliable. Further improvements can be made to the device in the blood pressure monitoring system. Care and safety to paralyzed patients. Again, it can be used for greater improvement. Thank you. Now, we have a video.
Thank you.
Hi team, thanks for the presentation. I have a very basic question, and I did notice the limitation of this system, that it cannot be used with someone who is fully paralyzed.
Yes, sir.
That leaves the other segment of the populace, which is partially paralyzed. Do we really need to do hand gesture? If someone can just it's so dexterous that they can turn their hand. Can this be solved using a button? Like we can just put 4 buttons and just send out the same set of signals. Is there an issue with such an implementation?
Actually, there's no such issue. There's no issue with the button system. This
We
was
can
just
make the
an
buttons
innovative
tactile
idea.
also. Okay.
Yeah, this was just an innovative idea of using hand gestures.
Okay. And at one point of time in the disadvantage itself, we had mentioned about massive data. So what kind of data are we sending from those hand gestures that is flooding the system? So you have a system that understands those hand gestures and then converts into a signal, right? So
yes, sir.
What's the format of data? Isn't that is a digital signal? Is it an analog signal that you are sending it to the processor?
Yes, sir. About the massive amount of data, if there are any further improvements to the device, like blood pressure monitoring or heartbeat monitoring, eventually the data accuracy should occur. And for the signal being transmitted,
So audio is not very clear. I couldn't decipher what you were saying. So your audio is alternating between coming and going. It's just like 4 times. I can hear that, but then it's just completely gone.
yeah. Am I audible now, sir?
Yes.
Okay. So about the accuracy issues, when there are any improvements made, the amount of data and there can be accuracy issues. To transmit it to the
Your voice
receiver.
is breaking, Meenakshi. Can somebody else answer from the group?
Actually, I've logged in through a single laptop. Am I audible now?
Yes, at times you are audible, but then it seems as if you are talking from far off from the microphone.
Okay,
Probably
yeah.
that's the issue.
So, like in future, when we improve the device, like adding this pulse monitoring and blood pressure monitoring, and we store the data. So obviously, the data gets increased, and this may cause accuracy issues or something.
Okay. Got it.
Okay. So my question is that a very good point was put up from one of the team who presented in the morning also, the similar sort of project. That why gloves? Again, my question is that if a person is paralyzed, then it can be a problem to ask them to wear the gloves, or it will be very problematic when they have constrained movement, and then by wearing a glove you are constraining it more.
Yes, ma'am.
Why cannot be a wearable?
Yeah, even I saw the presentation of that team. They did it with a watch. Yeah, so basically the watch will be wearing it on the wrist. So while moving, like we have to move the hand from the elbow till the wrist. Like even that will be a problem in moving. So the gloves is only for the palm.
No. My question was with them in the morning that why not wearable? My question with you is why not wearable?
The gloves is wearable, ma'am.
Yeah, no, not in a way. Why don't you think that we can make it in the form of watch? What will be the drawbacks?
A glove.
Okay. Yeah, and second question, guys, that you're using accelerometer, right, for sensing.
Yes, ma'am.
Am I audible? Am I audible to everybody else?
Yes, ma'am, you're
Yes.
audible.
Yeah. So the second question is, guys, that you're using accelerometers for sensing, right?
Yes, ma'am.
So what is the output you're detecting? What is the output you get from accelerometer? What is your serial monitor of Arduino is showing you in terms of reading?
It shows in a binary format.
No, that's fine. It is getting converted into digital format. It will be in binary. My question is that what is that end product? It is analog signal. It is some wavelength. It is some current, anything. What it is? What actually accelerometers converts, and what is the end value you get from accelerometer?
We get signal based on the gravity, like the
Gravity. Okay. Thank you. That's all, guys. Thank you.
Thank you, Team Health Engineers. Next, we have Team 21, Arrows, Aishwarya, Akash, Ridhima, and Samrit.
Yes, we are here. I'll be presenting the screen.
Okay.
Is my screen visible?
Yes, Aishwarya, you can go ahead.
Yeah. Yeah. So our project is to detect the vaginal infection, and we are 4 of us in a group. Our project guide is Jisha P, Dr. Jisha P. We're from BMS Engineering of college, and from the Department of Medical Electronics, 6th sem. Coming to a very brief introduction, what is pH? pH is a unit of measure which describes the degree of a solution which is acidic or basic. It is measured on the scale of 0 to 14. The vaginal pH pH is age dependent. The normal vaginal pH for a woman of reproductive ages ranges from 4 to 4.5, whereas for pre and post menopausal, it might be slightly higher than 4.5. The problem definition is that in many cases, the vaginal infection in women go undetected due to privacy issues. And also 3 out of 4 women today are facing this challenge. So we have come up with a solution in which we can test at our homes, and also it protects our privacy. So the methodology for this is that we are using a pH sensor kit, which involves a pH electrode as well as a sensor, which is connected to the Arduino. pH electrode is analogous in nature and therefore is voltage dependent. And we need to calibrate the sensor. So we have taken a buffer of known pH to calibrate the sensor. And we've also done the testing on various other solutions. In future, we would like to come up with a product. So this is how we would be testing our product. So first, for hygiene purposes, we'll have to wash our hands, and obviously everybody would have undergone corona test. So it's very similar to that test. Take a swab and spread your vulva and insert the swab and swirl it gently for 2 to 5 seconds. After 5 seconds, the swab can be put into a solution, a buffer solution. Akash will be continuing further.
Basically, there are two interfaces. One interface is between Arduino and OLED, and other interface is between Arduino and pH sensor. So in first interface, the VCC of pH sensor is connected to the 5-volt of Arduino, and the ground is connected to ground. Similarly, analog pin of pH sensor is connected to the A0 of Arduino. And in the second interface between OLED and Arduino, the SCL, that is clock line, is connected to the A4 of analog pin, and STA data line is connected to A5. And VCC is connected to the 3V3, that is the output of 3.3 volt resistor. As you can see, we have interfaced the model using pH sensor. I saw the next slide. So once the code is uploaded, the OLED display will start showing the pH values. So I would like to tell about the pseudo code now for Arduino. So we have defined the analog input and output, input for sensor and output for OLED. We will initialize the serial monitor and we will set the baud rate of around 9600. And we have taken values continuously from the sensor, like 10 values, and we arrange it in ascending order. And we have taken the average of those samples, and later we converted those values into the pH values using some equation. When the digital value is obtained, it is displayed in the OLED. As you can see, that pH sensor OLED display showing around 3.52 is for one of the sample. So first we insert the sensor into solution. If it is acidic, it will display on the OLED, or else it will check for the neutral. If it doesn't satisfy, it will check for base value, and it is displayed on the OLED. I saw the next slide. So the main advantage is that the device is user friendly and there is no complexity and it provides privacy to the users. The main disadvantage of our product is it is bulk in nature and it cannot be portable. So now I request Aishwarya to play the video of our project. So we can see the output for one of the sample, that is around 4.24, which is acidic in nature. And for another vaginal sample, it's showing around 3.78, I guess. Yeah. So hence we can say that it is acidic in nature.
Good afternoon. So after we developed a vaginal infection detector, we had to do the development of a chemi-resistive sensor for vaginal infection detection. Next slide. So what is a chemi-resistive sensor? A chemi-resistive sensors are materials that change its electrical resistance in response to changes in the chemical environment around it. They rely on the direct chemical interaction between sensing materials and analyte. The sensing material and analyte can interact by covalent bonding, hydrogen bonding, or molecular recognition. So the methodology we have used is conducting polymer-based electrochemical sensors were developed to measure the pH value of the vaginal samples in real time. The material was synthesized in acidic medium. Characterization of materials was done by SEM, that's scanning electron microscope, to check for the morphology. FTIR, which is Fourier transform infrared spectroscopy, was done once before and after to check for chemical structure of the material. The coating of sensing layer was done by drop casting or spin coating. After this process, we can detect if this specimen added is acidic, basic, or neutral using our developed sensor. Next slide. So the work done is, in the first step, we set up an ice bath using deionized water. Step 2, adding 1 molar HCl of deionized water. Step 3, stir for 15 minutes and add aniline. Step 4, add APS for chemical polymerization to take place. Next slide. Samrit will be continuing from here.
After chemical polymerization, we go ahead with the filtration process. The contents left on the filter paper can be seen after the filtration in step 5. And in step 6, we can see polyaniline, PVDF, that is polyvinyl iodine fluoride, and NMP, that is the binding agent. In step 7, we make a paste of these chemicals mentioned in step 6 with a 5:1 ratio of polyaniline is to PVDF. And in step 8, we coated this paste on a planar electrode and heated it using an hot air at 60 degree for 1 hour. Next slide. We then took pH tablets of pH 4, that is acidic, and pH 7, a neutral tablet, powdered them, and we checked for the resistance using nanovoltmeter and nanocurrent meter by dropping the solvent through a micropipette on the sensing layer. So we got the values, and we have observed that with the decrease in resistance, pH increases. Next slide. So the intentions of the product are to provide faster results compared to hospital, obviously, because if we use the vaginal infection detector, the patient does not have to go to the hospital. And this also improves the privacy of the patient, that they don't have to go to the hospital and it can be done at home. Third is it is cost effective and it can be available in medical shops as well. Next slide. These are the references. Thank you.
Hello guys. So I have one question. You mentioned that you're using for current prototype buffer. Can you tell me the concentration of buffer and what is that buffer?
Ma'am, we have used the ionized water, ma'am.
Okay.
Just for calibration, I have varied the potentiometer. So just to calibrate the sensor, ma'am.
Okay. Any specific quantity which needs to be used, which should not dilute it more?
Yeah, actually we used around 10 ml, ma'am.
Okay. Okay. That's all.
Thank you, ma'am.
Thank you, ma'am.
Shubham sir, do you have any questions for the team?
No, none from my end.
Okay. Moving on. Thank you, Team Arrows. Now we have Team 20, that is 4 a.m. Shashank.
Hello, am I audible?
Yes, you are audible.
I'll present it.
Yeah.
So, yes. So, wait, 1 second. So is my screen visible, guys?
Yeah.
Yes, your screen is visible.
Yeah,
Can
Krishna.
you proceed?
Yeah. So good afternoon to one and all present here. We are the team of 4 AM and we present you a product, Alveola, the intelligent mask. My teammates are Gagan, Mohit, Shashank, and myself, Sri Krishna. And our project guide is Dr. C. Gurraj from Electronics and Telecommunication Engineering Department, BMS College of Engineering, Bangalore. Next slide, please. So quickly, we'll go through the introduction. Yeah. So as per the famous Nostradamus predictions, COVID-19 happened in the year 2019, right? We had a survey in which we found that almost 33% of people were affected by COVID-19 and the others had relatable symptoms, but few also had a natural immune to the virus. We believe that prevention is better than cure, and that is why we have developed a prototype which can actually serve the society, and we have introduced this to the product, our product to the market. So next slide, please. Yeah. So quickly, I'll go through the problem statement. After COVID, everyone's major concern was health, especially lungs health. But there was no any existing technology or a faster technology to actually determine this lungs health. For example, for heart, we have smartwatch, which can give you a pulse rate or a heartbeat or anything like that. But for lungs, we don't have any existing technology or a faster technology. So that brings the significance of a project. So next slide, please. So as I told you, a project is a smart end-to-end prototype, which gives the output as lungs parameters. It defines lungs health, especially it defines lungs health. This is a basic product. Yeah. So coming to the uniqueness of a product. So our product it takes nasal inhalations as a basic parameter, basic input parameter upon which we define a sinusoidal wave, and we have also developed an algorithm which can actually intake the sinusoidal wave and it gives you the nasal inhalations as a result. And this product is most compactable and it is small in size, and mainly it is wireless. One more bonus unique parameter is that it can be implemented into a smartwatch. And now my friend Shashank will be continuing over.
So coming to the methodology of our project, as our friend mentioned about the nasal inhalation, we actually obtain our input with the help of a Mariota NTC sensor, which records temperature data set. And upon that, we have developed an algorithm based on a proven breath matrix toolbox in MATLAB. So upon that, we obtain theoretical formulas of lungs parameters upon which we define the lungs health analysis, and we present it in a visual analysis through a web app. So coming to the prototype of our hardware, we have actually integrated the thermistor in a mask wherein it is very close to the nasal inhalation of our breathing, which is our nostril, and that is converted to digital values with the help of an ESP32 module and transmitted over Bluetooth with the help of ESP32 module. And this is transmitted to the web app with the help of the Bluetooth. So coming to the flow diagram of our project, as I mentioned, the data is acquired from the ESP32 module and transmitted over Bluetooth to the algorithm, wherein we develop formulas based on the breath matrix toolbox obtained in the MATLAB. So upon that, we have done the result analysis through IEEE papers and internet standards, upon which we have visualized the data over a web app. And thus we have obtained our business model as well. So coming to the challenges we faced, since the nasal inhalation has a very noisy signal, we had to remove the noises with the help of filters. So in the device part, in the hardware part, we have actually implemented a low pass filter wherein we obtain a smooth, actually a fraction of smooth desired signal in the hardware part. And then in the software part, we have fortunately found a Savitsi-Golay filter in Python, wherein we can denoise the signal and have a smooth signal. And upon that, to detect the parameters, we had to bring it to scale it to a zero baseline, wherein we had to correct it to a zero baseline correction to obtain the lungs parameters. And coming to the implementation part of the algorithm, the zero values are extremely important because that is where we define the basic parameters of the lungs. That is the inhale and the exhale onsets, wherein it is the parameter at which the person inhales sharply is the inhale onset, and the person at which he exhales sharply is the exhale onset. So as you can notice, you have an orange mark, which is the inhale onset, and a green mark, which is the exhale onset of it. And upon this, we define the parameters. One such parameters is the breathing rate, wherein you define the number of breaths taken per minute in the human. And the other parameter is the average tidal capacity, which in normal terms would be your lungs capacity, which is actually calculated by the integral around the curve, that is the area around the curve. So using this, we have found the lungs capacity, that is the average tidal volume, which is the sum of the inhale volume and the exhale volume of a normal human. Now my friend will be explaining you about the result analysis part. Yeah.
So as you can see, these are some of the parameters out of from the result we provide. Starting with inhale onsets and exhale onsets, which are the starting points of the inhalation and exhalation from the waveform. Breathing rate, which is the number of complete inhalation exhalation cycles per minute. Average inhale and exhale volume is basically the capacity of the lungs based on inhalation and exhalation respectively. And the average tidal volume is the average total capacity of the lungs, and minute ventilation, which is the total capacity per minute. And coming to the normal range and the abnormal range, basically normal range, which we have defined, is based on the breath matrix toolbox, which my friend Krishna mentioned, which is the major reference for the project. So if the result is in the normal range, we have classified the result based on the values, if it is normal or abnormal based on the range. So now let's go through the video demo of our project. Video demo. So we provide the result in a web app, which is a 3-page web app, starting with the sign up page, wherein the user creates an account. So you can see the user will have to enter the email address, username, and then password, after which the user can log into the data acquisition page, wherein the user have to breathe for 2 minutes, where we collect the temperature data from the Maruta temperature sensor. So this is the timer page. So once we collect the data for 2 minutes, the algorithm works on the data to calculate the different parameters, which will be displayed on the third web app page. Just a second. Yeah, so these are the these are the parameters and their values. So if the parameters are in normal range, it will be displayed in green tabs, and if they are in abnormal range, it will be displayed in red tabs. So that is the basic idea of the web app.
Now coming to the application part of our project, this can be used in the fitness sector and as well as in the medical ICU treatments. Coming to the fitness sectors, nowadays people are very much people are very much consumed with the smartwatches, wherein we can also bring the mask and also present it in the smartwatches, wherein they can see the health analysis, lungs analysis through the smartwatches. And in the medical ICU treatment, as our friend mentioned, the minute ventilation parameter is one such parameters which is used in the ICU to treat any lungs parameters. So coming to the conclusion and the limitations part, not only our project is unique with the nasal airflow, but it is also very much user friendly when every user is respected by his privacy through his account, and he gets his outputs only in the same account. And this can be used in the further future enhancements to detect any lungs abnormalities, that is the diseases as well. And the limitations of our project is, since the recording of our data is very highly sensitive, we instruct the user to actually have a medical educated person to record the values, upon which the surveillance is taken, and then the data is recorded and displayed over the web app. These are references. Thank you.
Hi team. Hi Shashank.
Hi,
I have a very basic question here. What does 4 a.m. represent? The team name.
Krishna, I'll be explaining you of it. It's
Yeah, sir.
quite
So this
funny,
is
but
bring up to a funny matter. We believe that we as engineers, we work from 4 a.m. And we also had a synonym for this, 4 angry men.
Okay.
We are a team of 4,
Okay.
right? Yeah.
it's just a
Got it.
creative name of it.
Got it. After that, what are we essentially measuring using that system that we have put behind the mask? Is it the temperature or the airflow?
The airflow is recorded with the help of temperature, sir. When you actually breathe in, the temperature varies over the Murata sensor actually.
Okay.
It, As you inhale, the temperature increases in the sensor and it is recorded in the ESP module of it. So when you exhale, the temperature decreases. Upon that, we get a sinusoidal waveform pattern. Upon that, we are actually defining the parameters. One such parameter is breathing rate, which is essentially required for every human in normal day life.
So what are the temperature involved? Any idea on that part?
We didn't get you properly. Can you
So what are the temperature benchmarks? Like what is the
range
temperature
of the
of
temperature,
inhaled
sir?
here, exhaled here, right?
So the normal temperature, room temperature would be 28. So upon that, we have a inhale range of inhale peak of 32 degrees, and the exhale would be around 26 or to 24.
Is it? Shouldn't that be higher?
Sir?
I mean, if we are exhaling the air, our body itself is at 37
yes, sir.
degrees
But
Celsius.
in the Arduino code part of it, we have taken the negative part of it. So we have taken the contradictory part wherein the waveform is reversed to get the parameters.
Okay.
So there is this toolbox which I mentioned you about the breath matrix, right? So it actually has these formulas on the reflection or the replica of the waveform, sir. So upon that, the parameters are defined, or else you can't get the symmetrical kind of pattern, sinusoidal pattern in the normalization wave, sir.
And
So
how
that
do you
is-
detect if it is deviating from the normal pattern?
So as I mentioned about the result analysis, we have the set of ranges.
Okay.
Wherein it is proven by the breath matrix, by the Zeno labs. So they have done the survey of it and defined a normal range for rodents and humans, wherein we have taken the human part of it. So upon that, we are analyzing if the person has the values within the range, it would be shown in the green tabs for user-friendly application. And if it is in abnormal range, then it would be shown in red color, red tabs.
Okay. By the way, are you aware of spirometer? I mean, if we had
Yes, sir,
to just
the one
measure the
which
lung
you
capacity,
yes, sir.
right?
Yes,
You
sir.
can get that for $300 on Amazon, right?
Yes, sir.
And you don't need any professional.
Yes, sir. I agree with that. But in the spirometer, you actually define it through breathing of
Lung
it.
capacity, right?
Yeah, you take in the inhalation from your mouth in the spirometer, but our lungs is very much closely associated with the nasal inhalation, the breathing of through your nostrils. So that is very much close to your lungs. So you can detect parameters upon it, and this values which we have obtained is just the base for any lungs analysis. Upon this, we can actually have the future projections of disease classifications, or continuous monitoring would be like you can detect whether he's a passive smoker, or you could also give it in the fitness sector and help them also. So this is just the baseline of the technology which we are trying to bring into the market. The spirometer is already in one project. Yeah, Krishna, go ahead.
Yeah, sir. So our product is just a supplement for the existing technology. It's not as a substitute. So this is one point which we like to stress upon.
Fair enough. Got it. Thank you.
So guys, so I have one question. You are putting your basically temperature sensor in a mask.
Yes, ma'am.
Right.
Yes, ma'am.
So when you're putting it inside the mask, the temperature can vary because of the cloth also. right?
No, ma'am. I would like to object that if you closely
Have you
monitor
tested?
the. Yes, ma'am.
Have you tested the same thing?
Yes, ma'am. We have. So if you
Okay.
notice in this video part of it, uh, he is kind of like not very much close to the nostril, no, very much like accumulated or very much closed.
I understood, Shashank. There, my second question arises that once you add a electronic setup, little bit also, it will
Uh-huh.
have a certain weight. What's the weight of your mask after incorporating all these things?
Yeah, Krishna, please.
Yeah, ma'am, it'd be somewhere around few grams, ma'am, somewhere around 10 grams, because it's just an
See,
ESP32 mode. Yeah, ma'am.
because look it in a way that normally we don't like wearing mask, right?
Yes,
So
ma'am. Yes, ma'am.
you're adding some weight in it, so it will become
So
very
yeah,
uneasy and
I get your point. So sorry.
it will become very uneasy and uncomfortable for the patient, and that too if something is present in layer of the mask, it is touching your skin, there is an electronic setup, no matter how low you keep it, it will have a discomfort effect on the user.
Yes, ma'am. I would like to contradict that, and I agree with your statement as well, because everyone is like trying to get off the mask as well. So it's not just that we are making them to use it every day, or we don't want them to actually wear it and be continuously recording the data. Or it's just a device wherein in the crucial moments wherein he can preliminarily test his lungs or any such parameters by the mask. So I don't want him to wear along all the day. So we just want him to wear at the crucial moments.
Yes, but for the some time when you're saying, why I am telling you uneasiness, because you're saying it will be required a professional assistance while they are doing the
In the
test.
beginning stage, ma'am. In the beginning stage.
In the beginning
Yeah.
stage, let's see. The point here is that once a medical product comes out, okay,
Yes, ma'am.
how many times you train a patient, they will do some of the blunder. So as an engineer, we always say that it should be dump proof, right? So
Yes,
what
ma'am.
is the
Yes, ma'am.
guarantee that the patient will not start breathing from the mouth? There'll be
Yes, ma'am.
variation. How can you control that? So
Yes,
your
ma'am.
device should be smart enough. Listen. Your
Sure,
device
sure.
should be smart enough to show this error. Okay, there should be a temperature variance when you're getting a data from nasal and getting a data from mouth.
Okay. Ma'am, we
So
would
there
like
should
to take this as a suggestion and improve as well as
Yes,
this project.
Yeah.
because why I'm telling you this thing, I am currently working since past 2 years from on COVID particularly. So this can be very helpful for you, because I've seen even the nurses also, even the practitioners also, if the patient will do it twice or thrice, data accumulation wrong, in a wrong pattern, if your device is not smart enough, it won't be not be take it seriously.
Yes, ma'am. Yes, ma'am. I agree with your
And
statement.
second thing, the suggestion can be that you can look into the example for Philips. They come up with a mask.
Yes,
If you
ma'am.
see, the electronics is outside in a small embedded circle, which is incorporated outside the outer layer of the mask, not inside.
Yes, ma'am.
You have to find out some way that would be my suggestion, because it becomes very uneasy for a patient if you keep electronics inside.
Yes, ma'am. We have actually implemented a double layer mask wherein you get the
It's a
comfort
two-layer
of
mask,
the inner
wherein
line of it.
we need
If you want,
two
I'll
layers
show
of
the I mean, you could
the
like
mask.
see, no, ma'am. The mask is
I completely
yeah,
I
yeah,
got
yeah.
your problem.
Yes,
But
ma'am.
it
Yes,
just a
ma'am.
suggestion, it becomes very uneasy for a patient if something is inside. And second thing, the device or the mask should be smart enough to detect that what is the nasal temperature and what is the when somebody
Yes,
is
ma'am.
breathing
Yes,
from
ma'am.
mouth.
We would like
Because
to take a suggestion on it.
we all should think it from the patient point of view also. You no
Yes,
matter
ma'am.
how
Yes,
many
ma'am.
times you tell them, they will do it.
Sure, sure, sure. We would like try to implement in future now. Yes, ma'am. We would like to do that.
Thank you.
So that was it. Thank you, guys.
Thank you, ma'am. Thank you.
Thank you, ma'am.
Thank you, Team 4M.
Next, we have team 22. Thank you. Next we have team 22, team Blaze, Chandana, Likita, Sahaj.
Yes,
Is team
yes.
Blaze here? Yes, okay.
Unable to share the screen.
You aren't able to share your screen. Hello?
Yes, yes.
Yeah, so can you send it on the group? We'll share it for you. We are having some issues. Is Team Smart Triggers available? Team Smart Triggers.
Yes, ma'am.
Can you go first?
Okay.
To begin your presentation, yeah, please go ahead.
Yes. Myself, Steve Gladwin from Hinton College of Engineering and Technology. My project name is Demo X Cheto. Ma'am, 1 minute, ma'am. Let me check my internet. Ma'am, can you hear me? Hello.
Yeah, you're audible.
Yes. Myself, Steve Gladwin from Hindustan College of Engineering and Technology. My project name is Demo as Seto. The aim of project is hardware and software aim to design a smart remote accessible stethoscope. Our objective is by sending our heartbeat sounds via phone call to a doctor, and he can treat us within the remote accessible areas. Methodology we have used this. We have used some based SIM module, which is 2G. From that, we have inserted a SIM to it, and the phone number will be given to the doctor, and he can contact by that, and he can able to hear our heartbeat sound with the help of the stethoscope. The components used are condenser mic, Arduino Uno, and SIM 800 module, and battery, and wires, and software used is Arduino IDLE. This is a block diagram. When we call to that particular number, the SIM 800 get triggers and sends the signal to the Arduino Uno. From that, the mic gets activated, and from the chest piece, we can able to get sound of our heartbeats. This is my circuit diagram and the circuit connections. We have connected D2 of Arduino to RDXD of SIM 800L, and D3 to TXD of SIM 800L, and we have given a separate power supply to SIM 800L as it requires 4.4 volt batteries. Advantages: it is portable, and it is easy to use, and it is low cost. Disadvantages are signal may low at sometimes, and the SIM card balance cannot be found using this, and signal may low in some areas. Connectivity issues may be occur. Advantages: it can be used to save many patients, and it is useful for heart patient, and we can save doctors' life. In a communicable disease, the doctors communicable disease, when doctors goes near when a person communicable, the disease can be communicated to doctors too. At the time, we can use this as a solution. Future scope: it can be modified as a mobile phone. It will be very helpful to save many lives. And these are the pictures of my project. Conclusion: this can able to detect and help many doctors in the future. That's all.
Hi, team.
Yes, sir. Hi, sir.
Thanks for the presentation.
Yes, sir.
I'm just curious to understand, are we just talking about a stethoscope which is remotely accessible. That's the
Yes,
intent,
sir.
right?
Yes, sir.
Okay. Okay. And then by any chance, did you or do any research around what all tech exists as of now?
Sir, no.
Okay. Anything that is currently available and
Currently, it is available in Bluetooth modules, and Bluetooth type of stethoscopes are available, sir. And these much like of remote accessible from even anywhere from we can accessible, that like stethoscopes are not available at the moment, sir. And we
would
can
that
use
be a problem? I mean, if we can get the data from the stethoscope to some device via Bluetooth, we can always send it to any location across the world. Isn't that the case?
Yes, sir. But from this, we can modify it also as a mobile phone. Also, we can modify it, sir. So it will be very useful for any ways. From calling, also we can use for the health issues. For that, also we can use it.
And do you have a working prototype?
Sir, right now I don't have my working prototype, sir. I'm working on it because of some issues in that SIM 800L, and I need to rectify that. If I rectify it will be the working prototype.
So is it that it's not working now, or you are still working on making this work?
Sir, it's not working now, sir.
Okay, got it. Got it. So you have figured out the conceptual part of it and the components that you would be using, but the prototype is not ready yet.
Yes, sir.
Right. Okay. Got it. Thank you.
Thank you, sir.
Hello. I have one question, that what is the medium you're using to transmit the data? It's like a Wi-Fi module, something on cloud.
No, ma'am. It's like our phone calling, like that, ma'am. If we have coded that codes in Arduino, and Arduino will activate the SIM 800 module. In that, we have inserted a SIM, which the number will be known only to the doctor, and he can contact by that, and he can able to hear the sound of the heartbeats when we keep a set chest piece of the stethoscope in our chest.
Okay, so now the what I can see here is that you will be configuring only one mobile number with the SIM, right?
No, ma'am. The SIM will have the capable of receiving many mobile numbers, but the calling will be with only one number.
So the calling is what is important. No, your calling is a type of communication through which you're sending your data.
Yes, ma'am.
Yeah. So let's say I want to consult some other doctor. Then in that case, or let's say if that particular doctor is unavailable, then
Yes,
your whole
ma'am.
device is of no use.
At the time, the phone number, the SIM number will be known to the person, ma'am, the patient, ma'am, and they can say the number, while the other doctors can call to the number and they can able to hear. The incoming is many numbers, ma'am, but outgoing is only for one number. I mean, when we call from the SIM, it will be go to only one number, ma'am. If they call, we can attend.
That's my question, is that you cannot send it to any other doctor apart from that particular person.
Ma'am, if they know the SIM number and they can call to the SIM.
No, why a doctor? See, in most of the cases, a user will like to take certain consultations, right?
Yes,
if I'm failing some arrhythmia through which I want to get checked by any doctor, if that particular doctor is not available on time, then which doctor will be available in the world who will know that specific number to call you?
At the time we will be going to the doctor, and at the time we can inform them like that you can call to this and we can check that
So
you can.
that's the drawback, I guess, with the device. As
Yes,
Shubham mentioned, I guess exploring the Bluetooth can be a better parameter, because if you will use Bluetooth, at least you have a diverse connectivity. You can send that Bluetooth data to any doctor.
Yes,
There is nothing. Your service time will be less. Your you can open up with many more options.
Yes, ma'am. Later we can change it this as a mobile phone. If we convert it as a mobile, at the time we can contact many doctors at the case. Now this is a basic type, so it is configured only for one doctor.
Okay, so the other question is that is it a audio file, or which format of the file it is? What it is going to send to that doctor?
I mean, it will be sending our heartbeat sounds live capture. It will be capturing live sound of our heartbeat.
Now
and
let's
it
say
will
there
be,
is some connectivity issue, then the audio can be distorted also, no?
yes, ma'am.
There can be a possibility, because if it's an audio file, you're not downloading the exact audio file. It's a real-time audio file you are asking somebody else to hear out.
Yes, ma'am.
But you're connecting through a wire-based communication, basically, right? So let's say the connectivity where you are sitting maybe, or where you are sitting maybe less, and the doctor where he's sitting has more. So there'll be a lag in data. How will you cope up with that? It can end up in wrong diagnosis.
Yes, ma'am. I have no idea about it, and I need to think about it, and I will rectify.
Yeah. Okay. Thank you.
Yes, ma'am.
Thank you, team. That was an excellent presentation. Next, we have team 24. Sorry, team 22, Blaze. We have your presentation ready. Are you guys ready to present?
Yes, ma'am.
Okay. Are you able to view the presentation?
Yes, ma'am.
Okay, you may begin.
Yeah, good afternoon. My name is Likita and my teammate is Miss Chandana and Mr. Sahaj. And we are from ACS College of Engineering and we are the biomedical department. And this is our project called Arduino-based detection of kyphosis. And we are the under the guidance of project was under the guidance of Dr. Hanita Hess with a professor, Department of Biomedical Engineering. Then Arduino-based detection of kyphosis is nothing but kyphosis is the extended spinal curve, or it is also called as an hunched back. This is what the definition of a kyphosis. Next will be continued by teammate Chandana. Next slide, ma'am.
The slide has been changed. You can continue. Are you able to see it?
Yes, ma'am. Chandana, are you there?
Yes, yes.
Yes, please.
So the next slide, please.
Yes, they have sent already.
Okay. The next is the problem state. Sorry. Okay. The next is the problem statement. When I say the word kyphosis, that is interrelated with the spine and the posture. As we know, the spine is a backbone that gives the central support to the body that helps us to sit, stand, walk, twist, or bend. It plays a very important role in maintaining a posture. Posture is a way in which our muscles holds the body straight. Negligence towards maintaining this, it leads to many life-threatening conditions like lumbago, postural dysfunction, postural syndrome, etc. So there are mainly 4 types of postural dysfunction, which is also called as poor posture, which includes forward head, kyphosis, lordosis, and sway back. Among all of this, the mainly causes kyphosis because it was estimated that 540 to 560 million people are suffering from this kyphosis as the recent study. And in the figure, we can see that the normal spine and the kyphotic spine. So kyphosis, it is usually a hyperextended abnormal spinal curve that is at the thoracic vertebrae that is usually caused at the age of 40. This is most commonly caused for the women because of hormonal changes and menopause conditions. And it is mainly majorly caused in the adults. This is about the problem statement. Next slide. Okay, next is objectives. Here we are designing an effective method for the detection of kyphosis for continuous monitoring and correcting the posture. And we are using an Arduino, which is a microcontroller that is being used for monitoring of the kyphosis for different values of degrees and threshold voltages. And we are activating an LED light for any fluctuation that occurs in the patient posture. And we are using Arduino with the flex sensor to detect the bending position of the subject. And the system is going to create an alertness to the subject to correct their posture. Next will be explained by Likita.
Yeah, methodology. Methodology has 6 steps. First is the subject that has been interconnected with the flex sensor. Then it is connected with the microcontroller. It's in Arduino. Then processing will happen. Then microcontroller with the Arduino with the software. Then the output through the LED will be produced. When we talk about a subject can be a human being where you'll be having, it can be a sitting posture or it can be in the standing position. The posture where the spine will be occurred with the T1 to T12 where the kyphosis will be occurred. Through that it's called thoracic vertebrae. So from the T1 to T12 where the flex sensor will be placed, the flex sensor is also called as a bend sensor, or this is the flexible potentiometer. It sees the bending position of an human beings or any subject, that how much will bend. That is what the flex sensor will be see through this like it's a more flexible. And next, microcontroller Arduino we have taken here as an Uno. Then the Nano we have taken a Uno, and it is like developed by Arduino.cc. And this is what having an analog pins and digital pins and all in through this Arduino Uno, and this has been plugged through that. And processing has happened. All whatever the subject has been connected, then to the flex sensor, then to the whole the microcontroller, the Arduino, whatever the Uno we have taken. Then processing will happen. Then through this software only IDE we have taken microcontroller Arduino, that is the IDE is nothing but integrated development environment. Through that software, we have gotten source code through there, and we have been getting an output through the values. Then the output to the LED is like if it has been in the straight posture, is in a correct posture, the subject is, then it does not the LED does not blink. If it has been started to bend the posture, if it has been suffering from the kyphosis, it start to blink above 45 degrees, it start to blink, the LED will be. That's all about the methodology. Next slide. Yeah, it's a circuit diagram. We have been implemented with all the hardware and the software also. Hardware like we have taken Arduino Uno, flex sensor, resistors, like wires, jumper wires, and breadboard and all. And through this Arduino Uno, it was developed by like Arduino.cc, and it will be having some analog pins like 16 pins and all to be having. Then flex sensor, it's a flexible potentiometer, and these all the things. And resistor where it makes the current limiter, where it pull ups or pulls down these current whatever is coming. Then jumper wire, it makes in very much gold grips without any soldering and all. We can use this jumper wires and breadboard and all. Where this analog, the 5 volt is connected to the flex sensor. The Arduino, whatever is there, 5 volt is connected to the flex sensor, and there will be A naught. A naught is connected to the flex sensor of our next pin, and digital 13th pin is connected to the LED. That's positive of the LED. Then another digital ground, it has been connected to the LED of that same wire to the ground. Then analog pins all through the whole, it has been connected to the ground itself, to the resistor of first one. And software also we have been connected one pin, that is to the Arduino software. And this all been together has been connected to the human being with the flex sensor with thoracic vertebrae from T1 to T12. This all about the circuit diagram. Next slide. This is all about the project codes we have been used. And your 9600 is nothing but it's a constant value where the communication will be started from the software and the hardware. It begins there. That's all. Whenever if it has been lesser than 200 or greater than 200, we have taken here the degree, the value of it. So if it has been lesser than equal to 100, then LED start to glow and it will be very much high. If it has been greater than and equal to 200, then LED does not glow. It is in straight position. It's in correct posture. Then LED will be not blink or it does not any glow at all. It's all about the source code. Next slide. result from this all the project of the Arduino based detection we have found with the kyphosis that we have taken through T1 to T12 where thoracic vertebrae we have placed the flex sensor. Through that we have any state position is does not detect any light, LED will be not blink. If we started to bend above 45 degree, normal degree will be about 20 to 40 degree for a human being. If we started to bend till 45 degree over there, then it will be suffering from the kyphosis. So this has been resulted here. And if the flex sensor if it's been straight position is in when the flex sensor value will be 200, whatever value you have taken, that flex sensor will convert the value into degrees, that is 45 is in normal posture. If it is been less than 200 like 198 128 like that, then it's been greater than 45 degree, the flex value will be converted. So through the flex sensor on that time we'll be getting to know, oh, is in the kyphosis has been detected through this. So we can give a caution. They let the subject to be in a proper posture, not to be bent and to be a straight posture. This is what the result has been brought up from our project. Next will be continued by my teammate Sahaj.
Next slide. Advantages and application of our device. This device is used in physiotherapy clinic for detection of body posture. Can be used by the patients who are undergoing rehabilitation exercises. Helps in correcting the postures. Next slide. Next slide, please. Conclusion and future scope. This system provides early detection of kyphosis, which improve to preventive measures by correcting the postures. But due to advancement in technology, there is a change in lifestyle of the people in maintaining correct posture, which lead to many spine-related disorders. The developed system will improve the posture by detecting the kyphosis at early stages. Future scope of our device. This device can be developed for other bad postures like lordosis, swayback, and front forward headed, and also for any other part of the body. Next slide. These are the pictures of our project. In first picture, we can see that flex sensor is straight, but LED light is not glowing. And in second picture, we can see when flex sensor is bent, the LED light will start to glow. This will be same applicable for the subject when we place a flex sensor to the subject's back. Thank you. These are the references which we have referred.
Thanks, team, for the presentation. I'm curious to understand. I'll just quickly go through my questions. What was the reason for picking this particular project? Any interesting story around that?
Because nowadays everybody will be having a bad posture, not everybody having a laptops using and all, so that they may cause us with more often problems in the bad posture, and it can be lead for many other cases also. So it is more in the population, so we have taken this.
But isn't kyphosis restricted or prevalent in adolescent age itself, or is it universal?
No, it's in from adult also. If they are not in a good posture, it may attack for the adult people also.
Okay. And what kind of sensors are we using? Flex sensors. There are different kind of flex sensors. So what's the sensor that you have used?
Fiber optics we have. Many we have to the types of flex sensor we have. We are using only the just depend upon their heights also we have to use the flex sensors. Now we are using about 6 inches we have taken.
And I'm curious to know, I saw one of the screenshot wherein you had showed that when the flex sensor is bent, the LED glows. I mean, think about it. If you had to put that on some human's body, I mean, do we really need a sensor to do that? Why can't we just see and recognize that with the bare eyes?
For some certain,
sorry.
For some certain angles, there is a need for detection of it. For detecting that angles, we can help. We can use this.
But isn't that obvious from a physical diagnosis. I mean, looking at the person, and isn't that obvious?
Yeah, but in some who have undergone some accidents or what, they are undertaking the exercises. Then the doctor can identify the improvement by seeing the angles of day by day. So they can
Fair
use
enough. That's a valid point. Yep. So the data changes can be ascertained. And the size of that flex sensor, it felt isn't that too small to cover entire thoracic
just a sample.
vertebrae?
Yeah, it is just sample. For clinic use, we have to use bigger one, like 14 centimeter minimum. That it was not available, so we took for sample, small one.
Yeah, and to the point of what Likita mentioned in the beginning, I see a very huge market for such kind of a product. I mean, most of us would eventually be sitting at our desk and working on chair. So obviously, it can have a very huge potential in terms of like keeping our postures correct on a day-to-day basis. So I would recommend explore that area. That's why I asked, how did you come across such this problem statement itself? So explore that. It can be part of the chair, it can be embedded in a chair, and you can call it as a smart chair that recommends you. Sometimes it can even, nudge you or even give a little nudge by putting actuators that probably you should be sitting straight or something. So that can be a really good use case. That's all from my end. Thanks.
Okay, thank you, sir.
Hello, guys. I guess Shivam has covered all the points, so good to go from my side. Thank you.
Thank you, ma'am.
Thank you, team. That was a great presentation. Next, we have Team 24 Revolution, Vaibhavi, Anusha, Nidhi, and Vasudha. Are you guys ready?
Yes, we are ready.
Okay, you may start now.
Good afternoon, everyone. So myself, Vaibhavi, with my teammates Anusha, Nidhi, and Vasudha will be presenting our project, Development of Algorithms for the Prediction of Diabetic Charcot Foot. So we have worked under Dr. Joshi Manisha and Dr. Ajoy, who is an orthopedic surgeon and is currently working in MS Ramaya Hospital, Bangalore. So we are from BMS College of Engineering, Medical Electronics Department. So problem statement. So the disorder what we are working on is called as a diabetic Charcot foot, which is an irreversible medical condition in diabetic patients that leads to the weakening of bones. So initially it is characterized by inflammation, redness, swelling, gradually develops into joint dislocation, and also there are chances of ulcer formation. So and it is also characterized by high temperature and reduced loss of sensation. Gradually, they lose the sense of their foot. And if it is unidentified, it can lead to permanent deformation of the foot. So the first picture what we have put up is of you can make out the difference of the bone in a normal foot as well as a Charcot foot. So gradually this is what happens, and finally they'll lose their balance, or they won't be able to stand up properly. So, and there are chances also wherein subjects have to undergo amputation of the leg if it reaches a very bad condition. And so as you can see, it is characterized by high temperature. So this is what we are exploring on. So we are using a concept called as thermal imaging for the diagnosing of Charcot foot. So here you can, Anusha, the previous slide, yeah. The second photo, you can see the thermal images what we have put up. So this is of a patient who is around 53 years old. He has developed Charcot foot in a duration of 6 months. So his left leg, you can see that his temperature is 35 degree, whereas his right leg is just 31 degree. So there's almost a 4 degree difference, 4 degree temperature difference in his leg. So this is the area what we are exploring on, how to diagnose or detect or predict the chances of Charcot foot in a diabetic patient. Next slide. The objectives was to perform statistical analysis and develop novel machine learning algorithms to predict development of Charcot foot and to analyze performance and finally validate the results. Next. So the methodology that we have worked on is firstly data acquisition. So this was solely done by us. We have personally collected the images from 3 hospitals of around 62 subjects. So we have using these thermal images, we have obtained temperature values, and using the questionnaire, we've considered various parameters like what type of diabetes they have, the duration of diabetes, either if they have any kidney disorder or skin discoloration or pain, swelling, such parameters we have collected through questionnaire. So using all these, we've created a data set. So on this data set, we've performed 2 studies. One is statistical analysis and one more is predictive algorithms. So statistical analysis was performed on the software platform SPSS. So we have done 4 types of tests: Pearson correlation, student t-test, Welch test, and independent test. Whereas for machine learning predictive algorithms, we've done random forest, decision tree, XGBoost, and dialects. So next, my teammate Vasudha will be continuing.
Yes, next is methodology and data acquisition. We collected data of 50 diabetic and 12 Charcot foot subjects in 3 different hospitals using Fluke TIX560 infrared non-invasive camera. This camera captures normal and infrared images simultaneously. It has a 240-degree rotating screen to navigate hard to reach subjects. It provides temperature measurements of minus 20 degrees Celsius to plus 1200 degrees Celsius. We established the protocol for image acquisitions. At first, we took consent form from the subjects before the image acquisition. We collected the subject's details like name, age, contact number, whether they are normal or diabetic, if diabetic, duration of diabetics from each subject. The thermal images of feet were captured in dark room. The subjects were made to sit for 10 to 15 minutes before capturing so that foot temperature comes in normal range. Anything on the foot were set to remove. For example, socks, but if they're wearing lotion and all, they were set to wash, and if socks must be removed. And approximately 10 to 12 images were captured from different angles of each subject. The thermal images were analyzed using SmartView software. For analysis, we divided foot into 4 regions, that is front, back, left, and right. Maximum, average, and minimum temperature of each feet were noted down. And this is about methodology and data acquisition. Next. We performed 2 studies, that is statistical analysis, and we analyzed the performance of predictive algorithms. The statistical analysis was performed on SPSS software, which was developed by IBM. In statistical analysis, we performed Pearson correlation study, student t-test, Welch test, and independent t-test. Pearson correlation mainly measures the linear correlation between 2 sets of data, and the student t-test is method of testing hypothesis for unknown standard deviation. And Welch test is used to check whether the means of 2 samples are different or not. In independent t-test, means of 2 independent groups were compared. And coming to the performance predictive algorithms, we used Jupyter Notebook in prediction of algorithms, and the algorithms were random forest, decision tree, XGBoost, and dialects. Random forest was used to predict the possible outcomes for future, and decision tree is a supervised learning technique. and its tree-like structure shows us the prediction from input feature. And XGBoost, or it is also called as extreme gradient boost, works on the principle that is distributed gradient boosted decision tree. And DALX, it interprets the model and it predicts the outcome. Next will be continued by our team lead.
Coming to the implementation part, first we'll be talking about statistical analysis. This complete project took us around 10 to 12 months duration. So in this, the table that is shown here shows the parameters that is considered and temperature that is maximum, average and minimum for 50 diabetic subjects and 12 Charcot foot subjects. After the analysis, what we have got is that average age of 50 diabetic subjects is 56, and that for Charcot foot subjects, we have got 61. Next parameter that we have considered is duration of diabetes. That is from how long a person is suffering from diabetes. In this case, we have got 10 years for diabetic group and 15 years for Charcot foot group. Next observation is that Charcot foot group temperature is shown to be higher than diabetic subjects. There's approximately 1 degree increase in temperature in Charcot foot. 1 degree might sound very small, but in case of temperature, it is really high. Next slide. Yeah, this is one of the screenshots. The top one is one of the screenshots of the statistical analysis that we did on SPSS software. This is how we get the output. So in that, the positive correlations are the ones that have the double asterisk sign, like left 3 maximum 708.708 shows double asterisk. So here, as the duration of diabetes increases in Charcot foot group, temperature also increases. And now speaking of this tabular column of student t-test, two-sided p considered as 0.05 is the probability that the statistical test is satisfying the assumptions, that is the null hypothesis that we've considered. And 95% confidence level of the mean is a range with an upper and lower number calculated from our sample. This is because true population is unknown, and this range describes possible values that the mean could be. So this is only for the student t-test, but similarly we've done for all the tests. Next. So this is, it shows screenshots that we have got on Jupyter Notebook, and this is for the algorithm for machine learning. The first picture shows XGBoost algorithm, and here it shows which features are getting the highest importance. So here you can see right leg left region, maximum temperature has got the most importance. The second picture shows all the features that we have collected through questionnaire, and these we have used in the code. So like age, duration, everything is here. So here you can see we have considered the fifth subject. On the first line, you can see that we have considered fifth subject, and in the result we have got it as 0. So it means that the person doesn't have Charcot foot. And coming to the confusion matrix, we've got true positive as 12, true negative as 3, false positive as 1, and false negative as 3. To get even better results, we need to have we need to collect a larger data set. Next. So these are the results what we've got of statistical analysis. In the first study, correlation study, it was found that between duration of diabetes and area specific temperature value in left leg, that is front, back and left region in Charcot foot, there was correlation, positive correlation. And the next 3 tests, student t-test, Welch test and independent test, all all of them have the same null hypothesis, that is area specific mean temperature value for diabetic and Charcot foot study group is the same. That was the null hypothesis. And looking at the results, all of them had p-value greater than 0.05. So that means we have to keep the null hypothesis. Thus null hypothesis retained. Next. This was for the machine learning. These are the results. First, random forest sensitivity, we got it as 1. And R square value, we got 0.208. This is very close to 0. So that shows that the model is very poor. So we decided to do more algorithms. Then we switched on to decision tree. Here we got sensitivity as 0.22 and R square we got negative. So again, if the R square value is negative, it shows that the model is underfitting. So we can't consider that algorithm for our data set. Next, we went for XGBoost. Here, sensitivity, we got it as 0.75 and R square, again, we got a negative value, minus 1.04. Again, it was underfitting, but we got important features as maximum temperature of right leg, left region, maximum temperature of left leg, front region, and max temperature of left leg, back region. But these were the important features that we've got. Next, coming to dialects, we got sensitivity as 0.8. And important features as maximum temperature of right leg left region, maximum temperature of right leg front region, and max temperature of left leg left region. So out of all of these to consider, dialects was considered to be the best, and random forest and decision tree were ruled out. Next it will be explained.
Coming to advantages and disadvantages. Advantages, the camera we have used was portable and was easy to operate, and thermal imaging is a non-invasive technology. Whereas disadvantage, since the charcoal foot is rare, we got a small data set of charcoal foot and we require more data of charcoal foot subject to conclude it precisely. Future scope, it is required to follow up the diabetic subjects during regular intervals to understand the variation in the temperature. In case the person is showing any symptoms, the doctor will suggest their personalized footwear, which can eliminate the chances of causing charcoal foot. Limitation, we had very small and imbalanced data set. These are the parameters where we considered for predictive algorithms: age, duration of diabetics, type of diabetic, weight, HEBA, numbness, pain, swelling, deformation, skin discoloration, heredity, special footwear if any, kidney disorder, charcoal foot or no. The highlighted ones are the charcoal foot subjects. And we have used a SmartView software where we divided the foot into 4 regions: front, back, right, and left, to get the maximum and minimum temperature of each region. Conclusion, from statistical analysis, we conclude that there is a positive correlation between duration of diabetics and area-specific temperature in charcoal foot study group. From well-test, student t-test and independent t-test, it is concluded that the area-specific temperature for diabetic and charcoal foot study group is same. Coming to the predictive algorithms, dialect is found to be the best algorithm, as the sensitivity of the module is 0.8. And since we have a small data, we need more data set of charcoal foot study group, and it is required to follow up the patients of diabetic study groups to conclude it precisely. These are the references. Thank you.
Hi team, thanks for the presentation. In one of the last slides, you had mentioned a lot of parameters. How do you quantify parameters like numbness and pain? What's the scale that you are using?
So, so according to the doctor, when we consulted him, he told these are the more parameters which are the symptoms that a person developing charcoal foot might happen. So, right. There's no scale because each person describes his problem differently.
Precisely. It's very subjective.
Yeah, a pain which is more for me might be less for another person. But we have tried to understand when we were taking their images, when we were collecting images, we tried to understand, we told them to explain how, if it is severe or if they have continuous pain or if they have just like just once or just at random times. So like that we have categorized. So since there are previous researches on this, but all of them have used plantar images. But what we have tried is a very new technique. So so this is the new technique that we have used.
Sir, the thing is, like, we personally called them and we had to check everything. So what
Okay.
we did was we asked them from how long they have pain. For example, if they have some swelling, we asked them from how long are they facing it. And that's when, if, for example, somebody tells us they have it from few days or few weeks, then we considered it to be as no pain, because charcot foot usually happens after a long period of time. So if somebody tells it that they have it from 1 month or 2 months, then we considered it to be one. That's what we did, because this was a new project and we had to do the whole thing from like bottom. That is what we did.
Fair enough. Fair enough. Got it. Thank you.
Any more questions?
Hello, team. So I have just one small question. While testing your algorithm, what's the sensitivity and what's the specificity for your algorithm?
So sensitivity tells us, sensitivity tells us the if it's how much ever is detected is identified as yes. So yes, the disorder is there. So that is why we have considered in machine learning you can get many results like sensitivity, specificity, and there are precision, accuracy. But looking at us and doing some more research, we got to know that sensitivity will hold more value. So since it decides which disorder is yes or no, so we thought sensitivity will be the best parameter to assess the algorithm.
So how much it is right now? Maybe tested.
So so for dialects, for the method dialects that we've used, we've got 0.8. So among all four, that's the highest. But we can always try on more algorithms. We can, and one drawback is a much larger data set. So so that'll make a lot of difference in such projects.
Okay. Chances of error data. Let's say
There
you're
are,
giving the correct image, but let's say your algorithm is not able to recognize it. Any cases like that?
we tried logistic regression, so algorithm. We completely ruled out because it was not detecting any. So in 62 subjects, we have around 12 Charcot foot. So it was not detecting even one. So we completely ruled out logistic regression and we tried out the others.
Okay. So, and the third thing, the thermal data which you're taking, is there something which you're taking as the control parameter and the algorithm is comparing with that specific data set?
No, nothing like that. So what we are concluded from this is that temperature holds value in detecting or diagnosing this particular disorder. So as we showed, we compared lot of parameters, but in the end, we got to know that temperature holds a lot of importance in such cases for this disorder. So we can go ahead with this parameter. We can go ahead with this parameter to the doctor so that he can use this particular temperature value to diagnose this or to even predict it. So that is what we have proved from these tests. Yeah.
Nice. Thank you.
Thank you.
Thank you, team. That was an excellent presentation. Next, we have team 25, Ameya, Shruti, Vaishnavi, Anjana, and Vineet. Are you guys ready with your presentation?
Yes, we're ready.
Yes. Okay, you may begin.
So good evening, everyone. My team, Shruti, Anjana, Vineet, and myself, Vaishnavi, are going to present a project on deciphering emotional state based on facial expression. It is done under the guidance of Dr. Vijayalakshmi GV, who is Assistant Associate Professor from BMS Institute of Technology. So let me give you a brief idea of a project. Recently, during COVID crisis, quarantine and social distancing has affected the mental health of people. Emotions play an important role in detecting mental state of a person. They are directly related to the experiences faced by people. A person's body language, facial expression, as well as body movement, etc., come into consideration for recognition of human emotion. Mental disturbances can be diagnosed using facial emotion recognition. So emotions are often intertwined with mood, temperament, disposition, personality, etc. They are both the positive as well as negative parts of an individual. Both human emotions are often complex. So, like computers which perform quantitative tasks like addition, subtraction, they perform it very accurately and precisely compared to humans. But some other tasks like emotion recognition, which is performed more accurately and precisely by human compared to computers. So here our attempt is to make computer think and learn like human brain. In recent years, the advancement in machine learning has made computers recognize the emotions. A lot of research has been made where computers are allowed to recognize the 7 basic emotions, that is happy, sad, fearful, disgusted, surprised, etc. So in our project, we have trained models that can detect compound emotions. So compound emotions has two parts, that is dominant emotion and complementary emotion. So consider example like happily surprised, happily disgusted, or fearfully sad. In this, consider happily disgusted. In this, happily is the dominant emotion, while disgusted is the complementary emotion. But usually, compound emotions are harder to determine compared to 7 basic emotions. In this project, we have trained 4 predefined models and designed our own model. We have used here CNN architecture because it is usually preferred for face emotion recognition. So coming to our objectives, our project is to monitor the mental state of an individual with the help of compound emotions, uh, to examine and perform comparative study between different models, and critically review between different convolution neural network models. Our project is to come up with a model architecture that is simpler and easier to implement, to find the best fit model that has high accuracy and can be used to detect the emotional state of an individual. Further will be explained by Shruti.
So to come up with the methodology that we have used to come up with a model is being divided in 2 stages. In the first step for both of them was the data set collection. So we have used our images from the REF data set, that is a real world affective faces data set. We have taken 3164 images in the train set and 792 images in the test data set, which are equally divided and divided into 11 classes of compound emotions. So in the stage 1, we have compared the complex architectures of ResNet, Inception V3, VGG, and MobileNet, and another model that is another very basic convolutional architecture, which has only convolutional layers, only 2 convolutional layers, 2 mathful layers, and then 2 lens layers. So these are the 4 models are defined by transfer learning technique and compiled using suitable optimizers. In the when comparing the metrics of all these models, we have noted that a very simple architecture gives a very lesser accuracy of about 54% max. It doesn't cross that. Whereas the other models, which are much more complex, are giving much more accuracy than this model. But since the models are very complex, the ResNet architectures, etc., are very complex, we have our aim was to come up with a simpler model which can be better implemented in real time. So for developing our simple model, we have, for enhancing our accuracy of the simple model, we have added the feature extract handcrafted box features to the values that have been extracted from the previous stage to the simple model. So that increases the model's accuracy, as well as at the same time, the entire model is much simple to understand and easier to implement. So these are samples from our data set. We have categorized 11 compound emotions like happily surprised, happily disgusted, etc., as shown in the table. And these are some of the sample images that we have taken from our RAF data set. So the model architectures and implementation will be explained by Anjana.
So these are the 4 predefined models that we have used: VGG 16, ResNet 50, Inception V3, and MobileNet. VGG 16 is a 16-layer deep model. So the input to it is 224 cross 224 cross 3 image. and it has 3 sets of 3 convolution layers and gives an output. Coming to ResNet 50, this model was developed in order to completely reduce the vanishing gradient error. So this is done by using skip connections. Next is Inception V3. This has 48 layers, and the whole aim of this model was to decrease the computationality. So this is done by factorizing the convolution layers. Coming to MobileNet, this was done for embedded system applications. Even in this, the aim was to decrease the number of parameters, hence decreasing the computationality. That was done using depth-wise separable convolution, in which the image depth is convolved separately, and then it's combined using point-wise convolution. Coming to our model, this is the proposed model. Comparing the previous models in ours is pretty simple. It has only 10 layers. So the input of 200 cross 200 cross 3 is given to an input layer, followed by 3 sets of convolution layers and max pool layers. The output of the last max pool layers is given to a flattened layer. The flattened layer gives an output 1D vector, that is 33856 cross 1, which is given to the dense layer, and an output is seen. That is, the output is a 1D probability matrix. So what we saw is that this proposed model, which is 10 layers deep, gave a maximum precision and accuracy of 64%, which was not that good. Hence, we concatenated handcrafted features, as you can see on the right. More of this will be explained in the next slide. So our models have been implemented, trained, and compiled in Google Colab. So the features that we extracted, that is the handcrafted features, are hog features. It was extracted using the MATLAB software. So this features that is extracted is a 2D matrix of 3164 cross 4356 dimension. So this cannot be directly concatenated with the flattened output because the dimensions are different. That is 1D and this is 2D. So we had to flatten it. Next, we had to downsample it because they're of different orders. So we had to downsample it in order to match it with the order of the flattened output. Next, this downsampled matrix was of type CSV. So we had to convert it into a type Keras tensor, and then we concatenated it with the model that we had proposed. So after this concatenation, what we saw is that the precision and accuracy increased to a lot, and it almost matched with that of ResNet 50 and Inception V3. Coming to results will be explained by Vineet.
Yeah. So these are the results obtained by our model. The first graph depicts the precision. As you can see, the precision of our model increases with the increase in number of epochs. Same is the case with the accuracy, whereas loss decreases with the increase in epochs. Next, let us move on to the. So these are the emotions predicted by our model with our faces as the input. Next, the applications. A precautionary approach can be taken to deal with the meltdown crisis to warn caregivers through facial expression recognition. We can provide medicines to patients by recognizing their current state. Let us talk about the future scope of our project. Our project can be further extended to include detection from real-time video. We could have an alerting mechanism to alert caregivers about the real-time emotional state of an individual who is under surveillance. Number of classes for classification could be increased to consider a wider range of emotions. Now, talking about the advantages, our model has about 99% of accuracy. We can keep a real-time track of people to avoid situations of serious mental breakdown, where people tend to take drastic measures. The limitations include the time taken to train the model is very high. Models are complex and need CPU or GPU for implementation. Correlation between emotions and probability of an emotional breakdown could be improved. Now, the conclusion, all of our models were trained and a comparative study was performed. For 20 epochs, the accuracy of the hybrid model is about 99.95%. which almost matches the 99.85% of accuracy of ResNet 50, even though it has only 10 layers and a very simple architecture as compared to complex architecture of the ResNet model. The hybrid model can detect sudden emotional changes and can help in taking preventive measures to deal with mental health crisis. The metrics and the corresponding values obtained for the hybrid model are as shown in the table. Then these are
We
the
would like to
references.
show a real-time output for our project.
Okay, I'll share my screen. I hope it's visible. So this is the model architecture that we have defined. 10 layers with max pool and convolution layers finally flattened out. So coming to the predictions. So here is the number of epochs, that is 20, and these are the metrics values that is plotted. So this was an image given from the test data set. So 6 is the label. So 6 is basically sadly surprised. So it predicts as sadly surprised. And these were the inputs that were given. So what happens is here the input is given at set intervals of time. So here I have set it as 10 seconds. It can be changed by the user. So these were the inputs that were clicked at that intervals, and the emotion was predicted: fearfully surprised and sadly disgusted. I would like to show you now. Okay, so these were some of the images that were taken. This goes on until you stop the thing. So these were 3 of the images that were taken in real time, and predictions were made. So that's it from our side. Thank you.
Thanks, team, for the presentation. In one of the earlier slide, I noticed the term transfer learning. Where are we using that and why?
So transfer learning is the models that are already predefined are being implemented, like ResNet, MobileNet, VGG. So we have used those models to check the values that are given. Like we wanted to propose a model which is much simpler than all of these models. So to check to compare, we implemented these models first by transfer learning technique, and then these results we had compared with a simpler model, which was not very not matching much. So we improved our simpler model by concatenating the features.
Right. So during that step of concatenation, you had created hog feature extraction, if I'm not wrong, using MATLAB. Yes. So how did you identify those features? I mean, that's the crux of this entire project, that you were able to get better accuracy with less number of steps because you identified areas, a focus area on those images, right? So how did you identify those hog features?
So the hog feature that it gives the edges, like the direction of the edges. So that was extracted by using MATLAB.
Yeah, that is the tool. But how did you figure that out? Like these features should expedite the prediction.
So basically, we mostly aim to take the features of the structure of the face, basically. So we are dividing it based on the emotion. So emotion, it divides based on the structure, how the emotion is made. So sadly, fearful, happy. So all these features we found out that hog is the best feature to be extracted in order to take that structural part of the face. Hence, we used hog.
Okay, good. Got it. Thank you.
Thank you, sir.
Oh, hi guys. I have just one question. What's the cycle time? How much time does it take to once the user has uploaded the image? How much time it will take to give you the data, give you the result?
The result is almost instant. It doesn't take that much time. The only time it takes is to train the whole model, but the output, that is the prediction, is instant. It instantly gives the output.
Okay, that's all.
Thank you. Thank you, ma'am. Thank you, sir.
Thank you, team. Next, we have team 26, the three amigos, Anusha, Harini, and Harshita. Are you guys ready?
Hello.
Hello, yes, ma'am. This is Harshita.
Yeah.
I'll be sharing my screen, ma'am. Just a minute.
Okay, you may begin as soon as you do this.
Good evening, everyone. I hope my screen is visible and I'm completely audible.
Yes, Harshita.
Thank you so much, Anusha. Hello, everybody. Today we are here, three of us, team three amigos. Me, myself, Harshita Raju, and we are all from RV College of Engineering, studying in Electronics and Instrumentation Department, and our project guide is Dr. Anand Sir. So let me introduce my teammates, Anusha and Harini as well. Anusha, can you take a minute to say hi to everyone?
Yeah. Hello, everyone. I am Anusha. I'm from the same class, studying in 8th semester from RV College of Engineering.
And we have Harini.
Good evening, everyone. Myself, Harini. Even I'm in the same semester. I'm studying Electronics and Instrumentation in RV College of Engineering.
Well, great then. So today we are here to give us one presentation on our topic, segmentation of MRI brain tumor image using fuzzy field and k-means clustering logic algorithm. And let me begin with the slide. So as you can see in the previous slide here, we have a lot of terms, a lot of new terms like tumor, clustering, fuzzy field, all these may seem new to all of us, right? So in the introduction, we are here to tell you what exactly is tumor, what exact tools we are using in this project, and what exactly fuzzy field means. So basically, tumor is any kind of abnormal cells that may or may not spread in an organ. And based on the origin, there are different types of tumors like primary and secondary. Just a minute, I'll take a pointer. Okay. So there are primary as well as secondary tumors, and if they are detected in early stages, we can actually prevent a lot of invasive effects, and that is the main motivation of our project. And we have used the tool called image processing, which is one of the key subjects in our semester. So in this image processing, we have used it for the extractor and enhancing tool. And here we are creating 2D images, and those 2D image signals are then applied, and we are using the standard signal processing process over here, and then we are getting the result. And here for the image input, we have used the MRI, because MRI is detailed when compared to CET and CT. So we have another important term called as fuzzy logic. So this fuzzy logic is actually a mathematical tool which is used to represent vagueness. And application of fuzzy logic in medicine can be in PAD. So moving further, Looking at the problem statement, the aim of this project, it is known that in 2018, brain tumor, which is of fatal cancerous growth of abnormal cells in the brain, this is the most common kind of tumor among Indians. And according to IACR, which stands for International Association of Cancer Disorders, more than 24,000 people reportedly die due to brain tumors annually. And this is the most important motivation for us to start with this project. And that is also our problem statement, wherein we aim at producing MRI and brain tumor segmented images using K-means clustering logic algorithm. Here, MRI provides a very detailed picture of soft tissue organs like the brain, and also there's a lot of variation in intensity from black to white depending on the amount of fat and water present in each tissue. And hence, we have chosen MRI images as here. Also, the objective of our project, it is that the manual detection of the tumor in any of the like the manual detection is a cumbersome process. So our basic goal in this is to find out the segmentation through these algorithms. So here we have, I'll just brief you about the working of how exactly this process works. So here for the first one that we have for the C-means methodology, we have we need to first start initially by uploading the brain MRI image from the Google database or any other database. We got that. So into the MATLAB tool, and then after importing, we read the graphic image using the command. And here we convert this into the unit 8 to double form. This is done because basically for the image, because we use a lot of big numbers over here. So after so that's the reason we need to reshape it, and then it is converted from 2D array to a single dimension array. So then we actually select the fuzzy C-means and get the matrix. So what happens is that here, like after the conversion, the MATLAB function FCM, this should be selected by assigning the data and the number of clusters or the number of cluster centers, so that the FCM function will be converted to one-dimensional matrix array to four two-dimensional array. The thing is, what exactly happens is we find the center value of the we exactly find the center value of the cluster, and then we segment it accordingly. Here, since we actually made the two-dimensional arrays as one dimension for the easy calculation, here we do it, we reverse the process here again to find out the final answer, the final image, and then we have spatial functions which would be updated by the membership, and then we find the objective function. If the objective function if objective function difference is minimum when compared to the iteration, if it is yes, there's the end. That's the end of the process. If it's no, then the process begins over and over again. Basically, the clustering happens. So this is the code. I would show it. So basically, what happens is, if you look at clustering, so clustering is basically finding the middle of the basically if there is an image, there are different segments in an image. So we partition it and we find out which area has this proper tumor, which area is unaffected, what are different types of cells. So all these can be found out by using this tool. So that is what I just mentioned here. This is the code that we have written. So here you can see that if you upload an MRI image, our output with the segmented brain, segmented tumor, segmented bone, and the background. So these process done manually by experts or by doctors is tedious. So this is this whole processing by image processing is done through this program. So that is the crux of our project. And we have used another technique called as K-means technique. So in this also, we start off with the by updating the image, And then we check if the image is RGB, red, green, and blue. If it is RGB image, we convert into the gray image, because just to avoid the complexity and to increase the precision, because increasing precision is really important in this process. And then we convert that from 2D array to 1D array, simple, just to avoid the complexity and to get maximized results. And then we select K and get the matrix. Basically, we are trying to get the matrix. That's how it is differentiating between the normal cell and the tumorous cell, based on the intensity of the cell that is ranging from white to black. And then after that, we're finding the center value, and then we are reshaping the 1D array into 2D array again. And then we update the spatial member function just like we did in the scene. And here we are actually finding the Euclidean distance, the same that we use in our geometry classes in our high school. That is the square root of the square of the difference between the 2 points, subtracting the other two square of the two distances. So that's how we find the Euclidean distance. If that is minimum, yes, then that's the end of the process, that has reached the final thing. If the answer is no, then the overall process is cycling over and over again until there is a lot of precision, or until we find the maximum accuracy. So this is the code. Here it is the same. We are uploading the image over here, and then the image is getting segmented into 4 different blocks. So it would be really good if you can understand here, over here, we have this. So this is the input image, any MRI image that we have used here. And then this whole thing segments into segmented brain. So this is the brain area. So this is the tumorous area. So this is the bone part, and this is the background. So this is the output image of our process. So the same way, we have another example over here in which you can actually make out this is the tumor, but then you can also find out the tumor has spread over here, this part and the front part. So that is exactly covered over here, the segmented tumor, and this is the segmented brain and segmented bone. This is the background. So this is the result of a process. Similarly, we have used this. We have used a lot of processes, a lot of images, because just to make sure that the process is precise, the working is precise. So here we have used 2 techniques. So basically, one is scenes and the other one is scenes. So why exactly did we choose those 2, and which is precise and all that? Looking into that, we feel that scenes, in this scenes process, each element is assigned to all the available clusters with different membership degree for each cluster, and scenes cluster is a soft clustering method. Also, comparatively, this has better algorithm. So the efficiency of this algorithm is better. But one thing that is a disadvantage, I would say, is that it takes longer computation time. But comparatively, K-means, it takes a shorter computational time, but the algorithm is not that good. The precision is not that good, and it's a hard clustering method, and it is used for only single clustering. So we just went through the boundary and just use both of these algorithms. The advantage of this is that C-means and K-means clustering algorithms, they are best, the best algorithms you can say, comparative to any other algorithms, because of the reduced time. One thing is that we need to know the result as soon as possible, right? So that we can take immediate steps regarding the medication and everything. So that's the reason for the reduced computational time and clustering process. These algorithms are really good. Also, this method is automated, and it can help doctors overcome the difficulties of like minor mistakes that can be possible. So the main agenda of this project is to achieve higher and efficient diagnosis accuracy. So that is the overall advantage. The disadvantage, I could say that in case of C-means, the time, but it's not that time consuming, that I would say. So if we overlap these two, we get amazing results. If we overlap these things, we get best results. So looking at the future scope of our project, the future scope is that so this development of disease-specific application using this fuzzy. So this is the area of great research, and we can review, classify, and analyze, and go through a lot of any other cancers. Here we have taken specifically brain tumors or something. We can apply the same thing for any other type of tumors as well. That would be the future scope of our project. This could include lungs, liver, breast, any of those things. Also, the application does not only remain to the tumor, finding of the tumor or something. It can be to cure diseases like malaria, tuberculosis, or even asthma and diabetes, because we use a kind of multivariable control modeling for this specific thing. So that can be used. And one of the limitation that I could say is that there's uncertainty. So we are supposed to work on the precision that is due to the membership function. So that would be the limitation of the project. So here, this is a sneak peek of our simulation on MATLAB. And I would like to conclude that there are, as I already mentioned, that there are so many algorithms like region growing algorithm, hybrid clusters, and joint cluster. But we found out that fuzzy K-means or fuzzy K-means clustering algorithm, these are the best sort of algorithms for MRI brain segmentation, because of its accuracy and its precision and the way it is simple. So, so we would like to conclude that there have been several areas of focus where we have effectively solved the segmentation by using this FCM, and we have also uploaded a lot of different variants. So we have used the fuzzy C, which is very accurate and a bit time consuming when compared to K-means. But then we have chosen both the algorithms, and we have got results up to 93%, which is really a great thing. And HCM produces a close result to K-means clustering only, but it requires a bit of more computational time. That's all I would like to tell you. And here are a few references that we have used. Thank you so much.
Thank you, team, for the presentation. Let me take a step back here. If I have to summarize these last 17 slides, we are trying to compare clustering mechanisms or techniques, in particular, unsupervised clustering, right? And am I audible? Yeah, okay. And then we have FCM and FKM to compare against. We pitch them against the same data sets, and then we realized that FCM is more accurate, but the trade-off is more time is consumed. And then we concluded by saying that why don't we use both of them and get a better result, right? Okay, got it. Because it was a slightly detailed presentation to follow through. So I just wanted to understand if I've got the message right. Got it. Thank you.
Sir, I would like to share my screen and show it to you in real time. Is that okay?
Sure.
Like, is my screen visible?
Yes.
Yes, Anushka.
Okay. So this is the code that we have used. So like the tumor image that we are taking from the Google database, that I have named it as tumor 6. So this is present in my laptop, and like we have to also upload here in the MATLAB simulation environment. So here, like you can see when I run, the input image is there. One second. Yeah, this is the input image. Now you can also see the iteration counts. It is stopping at the iteration 67. Like as Irshita shown it in the flowchart, she said that whenever there is a minimum objective function difference, like whenever the difference between the last two iterations is minimum, then the clustering will stop, and like we can see the output. So like if you can calculate the difference between these two, like if you like 510 minus 503, if you do, it will be just 7, and 522 minus 510, it is like 12. So whenever there is a iteration difference is very less, till then it will keep on doing the clustering. Then only we'll get the output, like tumor background, tumor bone background, and the like image. That is what I wanted to convey. Like it mainly depends on the iteration count. And also you saw that like we were telling that the computational time in C-means is more. It is more compared to K-means, but it's not too more. It is like 5 to 10 seconds. That's all. It is more compared to K-means, but it's okay like for usage and all.
Got it.
Yes, sir. Any other queries, sir?
No. Sumati, do you have any questions?
Yeah, just one question that both the K-mean clustering and the other clustering, both of them will be using Fourier transformation to calculate your cluster value?
No, ma'am. It's different.
So there,
The K-means is
centered
Euclidean.
Okay.
Yeah, Anusha, go
Okay.
ahead.
Ma'am, there is a centroid formula and there is Euclidean distance formula. Like we have there will be one complete data set. Like if we give the clusters to be 4, it will divide the entire data set into 4 clusters. But in case of C-means clustering, each data point can belong to 2 or 3 clusters. But as in case of K-means clustering, each data point will become like will belong to only one cluster.
That's my question, is that in K-means it uses Fourier. It uses
Yes,
Fourier
ma'am.
transformation to get the clustering data compiling.
Yes, ma'am. Yes. Yes, ma'am.
What about the other one, C-mean?
Ma'am, like there are some other formulas, ma'am, like I can show you.
Okay.
Like there are centroid formulas, objective function finding formula, like some 3-4 formulas
That
are there.
it will not disperse your cluster point, it will keep them compiled together,
Yes, ma'am.
which will give you a better data.
Yes, ma'am. It always
Yes.
finds the minimum distance.
Okay, so the end
Like it will find the
vision.
minimum distance. Like after finding, if it is near to this center, it will put it in that cluster, or if it is near to some other center, it will put it in some other cluster, like that.
Okay, so you're saying the end product should be the your vision is to compile them together. So if you have to compile it together, what will be the points which you want to take from both the
Ma'am,
methods?
compile it together in the sense, we'll first perform FCM on the image. After performing FCM, then we'll do K-means, ma'am. Like not together, but first we'll do FCM and then K-means, like that.
So you'll opt K-means for the later product.
No, ma'am, we'll do both. Like first we'll do FCM, then K-means on the same trained results.
Okay, okay. That's all.
Yes, ma'am.
Thank you.
Thank you, ma'am. Thank you so much.
Thank you so much, everyone. Thank you for your time.
Thank you, team. That was the last team. So now we have a small announcement. The results will be announced via mail tomorrow. So we request you to stay in the group until you receive your results. I now request Jia Tomar to give the vote of thanks.
Am I visible?
Yes.
Thank you. Greetings, one and all. I, Jia Tomar, on behalf of the entire Medical Electronics Engineering Department of BMS College of Engineering, feel privileged to extend my hearty thanks to all those who have joined us today and those who have tirelessly strived to make this program, Biomed Technophilia 1.0, a grand success. A huge thanks to our revered principal, Dr. S. Murlidhar, Vice Principal, Dr. Bhimsha Arya, and Dr. Suresh Ramaswamy Reddy for their benign presence. Our sincere thanks to the distinguished chief guests of this national level project competition, Dr. Shripad Devala, as well as Ms. Sumati and Mr. Shubham, who have been kind enough to make some time amidst their busy schedule to grace this occasion. We are deeply grateful to our respected HOD, Dr. Manisha Joshi, our ever-supporting teaching and support staff, and other faculty members who have incessantly extended their cooperation, efforts, and support to all the students. Last, but certainly not the least, a huge, huge thank you to all the participants who have given their best foot forward in putting forth their ideas and on this wonderful platform. Wishing you all the very best. Signing off with a famous quote by the renowned American biologist Edward Wilson. In the attempt to make scientific discoveries, every problem is an opportunity, and the more difficult the problem, the greater will be the importance of its solution. So let's keep harnessing within us the curiosity and working towards building our ideas into a reality. Thank you and have a wonderful day ahead.
Thank you, Jia. I'd like to thank everyone once again for your participation and patient presence throughout the event. I hope you all have a great evening.
Thank you. Thanks, Pani. Thanks, everyone.
Thank you, everyone. Thank you.
Thank you, Sumati madam. Thank you, Shubham. Thank you. Thank you, student. And lastly, I'll thank all the student volunteers without which this event would not have been as successful. Thanks to all the volunteers who stayed from morning till this time. Okay, thank you one and all. Yes, we'll sign off for the day. Bye.
Thank you, ma'am.
Thank you, ma'am.